us off right
No. All right, it's recording.
Should we start?
Huh? Yeah, we can start, honestly.
You want to kick us off?
Yeah.
HTA?
Okay. All right. All right, welcome everyone to the exam 2 review. So just like this next slide is going to be basic logistics. Your exam is on Wednesday, obviously, from 6 to 7:20. Check your room assignments. If you have SAS accommodations, contact Professor Holmes. And the exam is going to cover anywhere from lectures 7 to 12, but it is fair game. I will say that if they ask you amino acids, that kind of stuff, it's like general things, not like crazy 1 through 6. That's all I'm saying, like not to worry you. The exam's closed book, obviously closed notes, same thing as last exam. And that's basically it for the logistics.
All right, so I'll kick us off by talking about carbohydrates. Just like a quick disclaimer, you have a lot of stuff on like glycolysis and pathways, and you need carbohydrates to really understand those pathways. But if we look at like what percentage of slides are dedicated to sugars, it's pretty small compared to the other content. So just this will be kind of like a quick overview. But yeah, usually for carbohydrates, you don't need to know like the nitty gritty. You don't need to memorize like every single sugar name. I will point out a few, but that's just a quick heads up. All right, so monosaccharides, I'm going to go into nomenclature. We have aldoses, ketoses, epimers, hemiacetals, ketals, anomers, and there's mutarotation, and then there's pyranoses and furanoses. So the general formula is CH2O parentheses N. So I'm sure y'all can figure that out. And then we have monosaccharides, so trioses, tetroses, pentoses, hexoses. And that refers to the number of carbons in the sugar. And then multiple monosaccharides, so we have monosaccharides, we have disaccharides, oligosaccharides, and polysaccharides, with di being 2 sugars, oligo being 3 to 10, and then polysaccharides being 10 plus. And these sugars are hydrogen bond rich. That's something really important to remember. And then they can exist in the linear or in the ring form, which I will go into later. One thing to note is there's stereochemistry, so there is chirality, so L and D isomers are enantiomers, and then glyceraldehyde, there's an example, so the OH furthest from the carbonyl is on the right for D and the left for L. It's kind of similar to amino acids, but if you can see that top figure, that shows like L versus D. And then D is almost exclusively used in nature, so you don't really deal with L, the L enantiomer. And then a molecule with N chiral centers can have 2 to the power of N possible stereoisomers. So that's a review of orgo. But yeah, something to remember. We have aldoses versus ketoses. So aldoses is when where there's a terminal aldehyde. So aldotriose is an aldose with 3 carbons. And we can see in the bottom left, we have like an aldotriose, or 2 of them, I guess. And then a ketose contains a ketone. So ketotriose is a ketose with 3 carbons. And as we can see here, the sugar has a ketone, not an aldehyde. So that's a big difference between the two. And then there are epimers. So structurally identical, except configuration around 1 carbon is different. Note that, so if we look at this figure, we can see that mannose is an epimer to glucose, highlighted by this red area. And then galactose is an epimer to glucose, highlighted by this red area. And so even though mannose and galactose are epimers of glucose, they are not epimers of each other. Because if we compare them, this at carbon 2, they're different. At carbon 3, they're the same. And at carbon 4, they're different. So that indicates they're not epimers. They're just stereoisomers. Are there any questions yet? Okay, nice. So we have anomers and mutarotation. So anomers are isomers that differ at 1 carbon, more specifically the anomeric carbon. And so here we can see we have an alpha anomer because the OH is trans to the CH2OH group. That's really important. That's how you define an anomer, is if the OH on the anomeric carbon is trans to this carbon number 6 CH2OH group. And then beta, they're cis. And then muted rotation is basically molecules can interconvert between alpha and beta anomers via linear form. So these rings are formed, and then they split up into a linear form, and then they reform. And when they reform, they can switch between the alpha or beta form just because of like SN2 or substitution reactions from orgo. But yeah, all that to say is that you have alpha and beta anomers. Oh, sorry. One more thing that's really important is The anomeric carbon always has 2 bonds to oxygen. That's how you identified it. It's the carbon that has 2 bonds to oxygen. So if we can see here, there's 1 oxygen and then there's another oxygen. That defines the anomeric carbon. We have the cyclic form, so pyran versus furan. So pyrans are 6-member rings, and the terminal OH acts as a nucleophile, where furans, we have 5-member rings, and the penultimate OH acts as a nucleophile. So if we have that linear form, the terminal OH will act as a nucleophile and form a puran ring, which is this top blue highlighted sugar. And then if we have the penultimate OH attack, we have a furanose form. And glucose is almost exclusively pyranose, but molecular vary in the equilibrium concentrations of each. So yeah, because it goes from linear to cyclized form, you may have an occasional furanose, but it's almost exclusively pyranoses. This is linear to cyclic form, just like how to do that. I think this is pretty important. Does anyone want to go over this? We can, but it's also in the slides if you want to go over it on your own time. No? Okay. We can. Yeah, go ahead.
Yeah. So I just wanted to say, there was an EdSTEM question on this where they were saying, like, what do the squiggly lines mean when you see this in diagrams? All it's just saying is that it mutarotates between the alpha and the beta, because what Ryan was saying, it cyclizes, then it goes back to the linear, and it cyclizes the other way. So the OH could go up or it could go down. That's all that these squiggly lines mean.
Thank you. Yeah, one more chance to say if you want to go over the slide. Okay, a few hands. All right, we'll walk through it. So as we can see, we have mannose. I'm just going to only really focus on mannose. So how many carbons? We have 6. We number it from 1 because this is an aldehyde, and so that's the highest priority carbon. We just go down the list. 1, 2, 3, 4, 5, 6. Then we draw a pyranose ring because here we're saying that we're forming a pyranose because we have the terminal OH attacking our aldehyde. You could form a furanose, but here for this purpose we're forming a pyranose. So we draw that ring. 1, 2, 3, 4, 5. Just memorize what that looks like, and you put the O at the top right. So number your carbons in this order, 1, 2, 3, 4, 5. Kind of just remember that this is what a pyranose ring looks like, and that's how you number your carbons. And then you can figure out, all right, where's carbon number 6? Well, connected to carbon number 5. So you draw the carbon number 6 above carbon number 5, and then you want to add your OHs. As you can see at the bottom right, this is really important. If we have a Fisher projection, which is up here, if your OH is on the left side, that means that your OH will be up on your cyclized form drawing. That's something really important to note. If it's on the left side, it'll be up, and if it's on the right side, it'll be down. So as we can see here, on carbon 2, the OH is to the left. On carbon 2, that would mean the OH is up. And then carbon 3 is the same, so it's on the left and it's up. And then carbon 4 is down because it's on the right. Are there any questions? Yeah.
How do you know where the OH on carbon 5 is?
So the OH on carbon 5 is this oxygen right here, right? Because this is doing the SN2 reaction. So the OH on carbon 5 is always at the top right of your pyranose ring. Does that make sense?
Yeah, I mean in the linear form.
Oh, so that's a good question. You need to know whether you have D or L. Like it has to specify whether you have a D sugar or L sugar. And then from there you can put it to the right or the left. Any other questions? Yes.
Which sugar should we be memorizing?
I'll get to that later.
We have in the slides.
And then, yeah, so then finally, if they specify, so here they're asking for the alpha form, we're just going to put our OH on carbon 1 trans to this CH2OH, or like carbon 6, as I mentioned. That's just the definition of an alpha cyclized sugar. This is a summary of that slide. It says the exact same thing, so I will not be going over it. So, segue is sugars to know. I will note, my year, I took this class 2 years ago. I memorized all of these sugars, and none of them showed up. With that being said, you should still memorize them, but just know there's a chance that you won't be tested on it. But yeah, the ones in the red boxes are the ones you need to know. There's a repeat, so D-erythulose is the same here as it is, or actually this is different. But memorize everything in the red box. Do you want to say anything?
There is a way to memorize it. This is what Sarah Taylor did 2 years ago when I first learned it. She said like glucose is going to be like the same as like basically flipping someone off, where your middle finger is going to be the OH sticking out, and then the rest of them are going to the right. So if you do that to yourself, you'll see that they actually align with the glucose things. And then it's the same with fructose, except we know fructose is actually a ketone. So that's the way I memorized it. I don't know, it was super salient, so I was like, oh, this is easy. And then with mannose, it's your, what's it called? Your index finger, and then galactose is your ring finger. That sticks out. But they're epimers of glucose. So if that flew over your head, don't worry, but that's just an easy way to memorize it. I can go over it after this session. Just write down a note on the slides, and I'll go over it again.
Yeah, that's also why I memorized it. All right, so just a summary of monosaccharides. How to tell. I will be reading off this slide. D versus L. So the highest number unique carbon OH, right D, left L. So yeah, the highest number carbon, not the CH2OH. So if we go back here, this is the highest number carbon. That's not CH2OH, and this is chiral center. Wherever that OH is, if it's to the right, it's D. If it's to the left, it's L. That's all that's saying. We have aldoses versus ketosis. So Fischer, the external aldose versus internal ketose carbonyl group. Yeah, ketone versus aldehyde. Hawthorne, Haworth, 1 CH2OH in ring, aldose. If you have 2 CH2OH in ring, you have a ketose. Epimer versus anomer, different by 1 carbon versus different 1 carbon at anomeric carbon. And then anomers are special epimers, so they're not mutually exclusive. Alpha versus beta anomers. Anomeric OH is trans for alpha and cis for beta to the CH2OH group. Furan versus pyran. 5-member furan, 6-member pyran. Reducing sugars. So this is important that wasn't talked about too much, is that the anomeric carbon has a free OH. If that's the case, it's a reducing sugar. If it's not, if that OH is in a, let's say, glycosidic bond, it's a non-reducing sugar. And we will go into glycosidic bonds in a little bit, but an alpha glycosidic bond is when the 2 lines that connect the oxygen between the sugars, when those lines are opposite directions. So one goes down, one goes up. And then beta is when they both go down or they both go up. But you'll see an illustration of that. Any questions on monosaccharides? All right. Disaccharides and polysaccharides. So disaccharides in glycosidic bonds, polysaccharides like starch, glycogen, cellulose, chitin, bacterial cell walls is peptidoglycans. Okay, so the formation of a glycosidic bond is a condensation reaction with a hemiacetal and an alcohol. So as we can see in the top of this diagram, we have a OH, which is highlighted, and then a hydrogen on this alcohol. And this is a dehydration reaction because we're getting rid of water. This reaction occurs, and then water is released, and now a glycosidic bond is formed. As we can see, there's one bond to this oxygen and another bond to this oxygen. Can anybody tell me what bond this is? Is it alpha or beta? Wait, say it again, sorry. I don't even know. Is it beta? Is it alpha?
No, that's...
No one? Okay, it's an alpha glycosidic bond. Does that make sense? Does anyone have any questions? No? Nice. Okay, so naming of glycosidic bonds. This gets pretty nitty gritty. I'm gonna just quickly gloss over it, but it's not super important. We have first sugar, so alpha or beta is a D or L, ring size, and then sil suffix. So as we can see in this first sugar, it's beta because the O group is up, so it's cis to the CH2OH group. So beta D, galacto, which is the name of sugar, purinosyl, which is the type of ring, and then we have the sil suffix. Then there's the glycosidic bond, which is a beta bond here, beta 1,4. And then we have our next sugar, which is the same nomenclature. So we have beta, because this OH is cis to the CH2OH. And then we have beta D, gluco, which is the type of sugar, and then pyrinos, which is the ring size, as well as os, because it's a reducing sugar. Can anybody tell me why it's a reducing sugar? Yeah.
You have a free anomeric carbon.
Exactly. Yeah, exactly. Free anomeric carbon to do other reactions. And then this I will not be going through, but it's the same nomenclature and naming process. So yeah, reducing sugars have a free OH, making them more reactive on the anomeric carbon, like we just said. So that's important to note. Reducing sugars are reactive. Non-reducing sugars do not have a free OH, so they're less reactive. And then this is just this is depiction. This is a reducing sugar because we have a free OH. This is a non-reducing sugar because our anomeric carbons are actually involved in the glycosidic bond. That's important to note. Your anomeric carbons, if they're both involved in the glycosidic bond, will not be reducing sugars. It will be non-reducing sugars. Okay. Any questions on that? Fantastic. Here are polysaccharides basics. So they're linked with glycosidic bonds, which is what we just went over. They can either be linear or branched. And then if they're all the same sugar, they're homo. And if they're different sugars, they're hetero. So we have homopolysaccharides. That first yellow string is unbranched. Then the next one is branched because there's a branch. And then the next one is heteropolysaccharide because that's unbranched because it's linear. And then the other one is heteropolysaccharide that is branched. And then different polysaccharides of glucose. So one thing to note is you have like 4 different types. You have cellulose, which is seen in plants. They use beta glucose and they have a beta 1,4 bond. And more specifically with beta, because the bond is opposite, it's able to form linear formations. So with beta bonds, you can form these linear formations. And that's why the shape of cellulose is in this very neat line structure, very difficult to break down. If we have starch, we have amylose, which is seen in plants. Here we have alpha glucose, which is an alpha glycosidic bond. Branches, no. But as we can see, it is in this like cyclical formula, this helical formation. And that's because of the alpha bond, because the bonds are both in the same direction. And that prevents this linear formation. If we go to amylopectin, we have plants alpha glucose. The bond is 1,4 and 1,6, which means there is branching. So as we can see, it branches. And then glycogen, which is in animals, alpha glucose, pretty similar to amylopectin, except here it's in animals. And then we have this branching here. So yeah, energy, which has alpha bonds, is in the form of starch in plants and glycogen in animals and bacteria. And then for structure, which has beta bonds, that's seen in cellulose plants, and then chitin, which is exoskeletons and cell walls. Do you want to add anything?
Just a quick note. Note that the non-reducing end right there of the alpha 1,6 branches of amylose, amylopectin, when we're taking off the glucose monomers, they come from the non-reducing end, not from the reducing end. I'll repeat that again. On the left side is the non-reducing. That is where we take off the glucose monomers if we're breaking this thing apart. Whereas if we're adding, we add them to the reducing ends. So just something to keep in mind and separate it is if the professors ask you like which side of, is it a reducing side or a non-reducing side that ends up breaking the glucose monomers from the polymer, you would have to designate which side.
All right. So I think the last slide of lecture 7 is metabolism. I think this is more of just like a soft launch into the next 3 or 4 or 5 slides, or I mean lectures. So catabolism is the breaking down, so generates energy. Anabolism is building, I don't know if I said that right, and it requires energy, so you're building things up. Energy coupling is how all of the pathways we've been studying works, which is standard free energy changes are additive. So we have second reaction has large negative free energy. It drives the first reaction. So this diagram does a really nice job illustrating that. If we have reaction 1 and it's uphill, and we have reaction 2, which is really downhill, we can couple these 2 reactions to make reaction 1 almost favorable. And so as we can see here, delta G is pretty negative, which helps make reaction 1 favorable. And then as we can see, reaction 2 is ATP. So we can say ATP hydrolysis is coupled to glucose phosphorylation. Basically saying we use ATP hydrolysis in a lot of processes to help drive them forward. Any questions on sugars? Nice. Okay, lecture 8. So bioenergetics and glycolysis. So this was like my least favorite diagram in this class. So I'm going to try to explain it. But redox reaction is the transfer of electrons. So oxidation is the loss of electrons, and reduction is the gain of electrons. Really nice tool is oil rig. So oxidation is loss of electrons. Reduction is gain of electrons. I've always used Leo Gur, like Leo, the line goes Gur, Leo, so loss of electrons, Gur, gain of electrons. You pick which one's better, but you know which one's my favorite. Reduction potential in V, so it measures the electron affinity. Oh boy. Okay, so higher electron reduction potential means more electron affinity. And so that's relative to hydrogen. And so electron affinity basically indicates whether or not a molecule will be reduced or oxidized. the higher the electron affinity, the more likely it'll be reduced. So oxygen, very high electron affinity, it wants electrons, so it'll probably be reduced. While if we look at like fumarate, not so much. Yeah, that's rough. Delta E equals E prime, reduction potential of the molecule being reduced minus the reduction potential of the molecule being oxidized. So if a molecule is being reduced, you take that value and subtract it by the value of the molecule being oxidized. Think of it as a summation of half reactions, but reverse the sign for oxidation. That's important. You have to reverse the sign for oxidation. And then to figure out spontaneity, we have delta G equals negative NF delta E. So N is the number of electrons transferred. F is Faraday's constant. Delta E is obviously reduction potential, but it's important to note that a positive delta E results in a spontaneous reaction because of this negative sign here. So that's important to know. If you have a positive delta E, you have a spontaneous reaction.
I was just writing the flipping from the table. So on the table, it's reduction potentials. You will have to flip one of them if you're calculating this. Now, if they give you the oxidation for one of them and they give you the reduction for the other, you don't have to do any flipping. So I was just writing as a reminder.
Facts. Okay, so oxidation of carbon. So the atom that is most electronegative owns the bonding of electrons. So increasing electronegativity, H is less than C, which is less than S, which is less than O, which is less than N, which is less than O. So O is the highest electronegativity. And if you have two atoms that are identical in a covalent bond, they share the bonding electrons. In other words, count all the electrons in the bonds to less En atoms. So example C to H. Count half of these E's in bonds to the same atom. Example C to C. Don't count E's to more electronegative atoms. So basically what this is saying, and the diagram to the right does a good job explaining it. I remember I was studying for the MCAT and I watched the organic chemistry tutor. He has a really good video on reduction and oxidation, or calculating oxidation states. But if we see this carbon in CH4, it has 4 bonds to hydrogen. If we draw those bonds out as 2 dots, which represents the 2 electrons in a covalent bond, carbon is more electronegative than hydrogen. And so each of these electrons surrounding this carbon are owned by this carbon. So that means our oxidation number for carbon in the CH4 is 8, and the oxidation number for the hydrogens is +1. Right? Yes. Yes. No. The high oxidation state of hydrogen is +2 because they get no electrons and then carbon gets... Wait, is that right? So if I have CH4, what's the oxidation state of the hydrogens? -2.
Yeah, it would be -2.
Okay,
Carbons
yeah.
are 8.
-2. Because carbon's oxidation number is 8 for the reason I explained and the reason that this slide explains. And since this molecule has no net charge, it has to be canceled out by the oxidation number of our hydrogens. So since we have 4 hydrogens, their oxidation number is -2. Yeah, do you want to say something?
Also, if you want to just be safe, you can literally just write carbon owns this amount of electrons, hydrogen owns 0 in this case for CH4. You can just write that to be safe. That way you don't have to deal with oxidation states.
Yeah. And then, so if we look at the next example, or let's take ethene, we see C bonded to C, so there's a double bond. Obviously, C will take the electrons from hydrogen, but the bond between carbon and carbon, they share the electrons. So even though there are 4 electrons bonded between 2 of them, each carbon will take 2 because they have the same electronegativity. And then lastly, if it has a bond to oxygen, as we can see in acetone, our carbon in the center that's highlighted in magenta shares 1 electron with the carbon adjacent and the 1 more electron to the other carbon adjacent. So it has so far an oxidation number of 2. And since it's bonded to oxygen, it's bonded to oxygen. Oxygen will take all the electrons, leaving our oxidation number at 2. Does that make sense? Does anyone have any questions? I think it's good to practice this, really draw out your electrons. And if you're confused, ask us. Oh, yeah, what's up?
So it's not really an oxidation number like as we learned it in like orgo. It's just like the number of electrons that the atom owns. It's weird.
Yeah, essentially. Yeah, you can, again, the safest way is just to write carbon owns this amount of electrons. Hydrogen owns this 0. Yeah.
But yeah, if you're confused, go ask us, post on that email, or watch the Organic Chemistry Tutors video, because that was really helpful. Okay, my favorite pathway, glycolysis. All right, so the first stage of cellular respiration, so glyco means sugar, lysis means splitting. The location is the cytoplasm of the cell, which is really important to note. It is the cytoplasm, not the mitochondria. It creates energy in the form of NADH, which will be used later in the citric acid cycle, as well as and the ETC, and also just makes direct energy in the form of ATP. It's anaerobic, so no oxygen is used yet, and there's two phases: the preparatory phase, where you use ATP, and the payoff phase, where you make ATP and NADH. And an overview of glycolysis. We have the preparatory phase and the payoff phase. The net equation is seen at the bottom: glucose plus 2 ADP plus 2 inorganic phosphates plus 2 NAD plus makes 2 pyruvate, 2 molecules of ATP, 2 molecules of NADH, 2 molecules of H2O, and 2 hydrogens. Note that this is the net equation, not the complete equation. That's important to note. And then just lastly, for your exam, you need to know the name of every molecule and every enzyme in this pathway. That's really important. You don't necessarily need to know the structure, but it is extremely helpful in your memorization, and it may be pretty helpful when you're answering the questions. Yeah.
I will say one note. I took this class last year. One of the sections was they did give you like the structures, and you had to like identify them, as in like write their names down. So you don't have to know how to draw the structures, but do have a general idea of what they look like, just in case. Yeah.
That's fabulous advice. Okay, so we have the preparatory phase, steps 1 through 5.
Question?
Yes. Oh, yes.
Question for the next question. Can you explain the 2H2 and 2H in parentheses?
That's just the amount of water that we generate from the enolase step, because we have two glyceraldehyde phosphates going into it.
Yeah, and the 2 hydrogens come from step 6. But yeah, we're not too concerned with water and hydrogen because there's so much of that in the cytoplasm that it doesn't really affect, we're not too worried about it. Any other questions? Okay. So step 1, what happens? We add a phosphate using ATP. So our equation is glucose plus ATP makes glucose 6-phosphate and ADP. And the reaction is seen in this beautiful diagram. And the enzyme is hexokinase. I'm sure you may have heard it already, but a kinase is something that transfers a phosphate to a molecule. And so for glycolysis, we're usually using kinases. We're using ATP with kinases, or we're making ATP. So if you have any idea of if you're using a phosphate, if you're using ATP, the name of your enzyme is probably blank kinase. That's something really easy to remember and helps you for memorization purposes. It attaches a negative charge to glucose. As we can see here, there's a very negatively charged phosphate, and that traps glucose in the cell because charged particles do not easily diffuse across the cell membrane, which is a lipid bilayer. So that's important to note. This first step phosphorylates our glucose and traps it in the cell. As we can see, it's extremely exergonic because we have a very negative delta G. And this is known as the first priming reaction because this is the first reaction using ATP. There's regulation, so glucose 6-phosphate acts as an inhibitor. Can anyone tell me why we would want to regulate this step? Yeah. Exactly. If we have very limited ATP and we don't regulate this step, and then we use ATP to phosphorylate glucose, we may die. So that's not good. All right. This slide is just a summary of everything I just said, so I'm going to move on. Step 2, what happens? We have a 6-member ring, and it turns into a 5-membered ring. So we have glucose 6-phosphate, and that turns into fructose 6-phosphate. The enzyme is known as phosphohexose isomerase. Phospho, because we have a phosphate group. Hexose, because we have a 6-membered sugar ring. And then isomerase. Can anyone explain to me why it's isomerase? Does anyone have like a memorization trick? Yeah.
Switching isomers from 6-member
Yeah, exactly. Yeah, so isomerase, that enzyme name is typically when you're changing isomer forms. So we're not adding anything, we're not removing anything, but we're changing from a 6-member ring to a 5-member ring. So isomerase, if you want to memorize it, just know it changed isomeric forms. It makes structures symmetrical and exposes C1 for phosphorylation in the next step. This is important because we can see C1 is anomeric carbon, but it's also like in this ring. In this step, we're exposing our carbon 1 to be phosphorylated in the next step. And yeah, delta G is like pretty close to 0, so we don't really worry about it. Step 3. Okay. What happens? We add a second phosphate group to carbon 1. It's a very exergonic reaction because it's coupled with ATP hydrolysis. As we can see, we use ATP, so our reaction will be very exergonic. And the equation is fructose 6-phosphate plus ADP makes fructose 1,6-bisphosphate and ADP. And our enzyme is phosphofructokinase 1. So as we can see, we're using ATP again. We're going to have the name kinase in there. And this is the commitment step. So fructose 1,6-bisphosphate will become pyruvate. There is allosteric regulation. So with this enzyme phosphofructokinase 1, it can be activated by AMP, which indicates low energy levels, but it can be inhibited by ATP and citrate because those indicate you have high energy levels. And if you have high energy levels, you don't want to waste energy making more energy, so you'll inhibit this reaction. And ATP can actually be an allosteric, can bind to the active site or at an allosteric site to help inhibit this enzyme. And then why would you want to regulate this step when the first step is already regulated? Can anyone explain that to me? Our first step is regulated just to preserve energy. So why would we want to regulate this step? Yeah.
The citrate regulation matches the glycolysis speed to the speed of the TCA cycle.
That's a good one. Yeah. That's a good one. So if you have if you're making a bunch of ATP or a bunch of pyruvate to put into the citric acid cycle and it can't keep up, you're going to want to slow down glycolysis so your citric acid cycle can catch up. That's a good one. Does anyone have any other ideas? Yes.
You could feed in at a different point so that you get past the first step.
Yes. Yes. You can definitely feed in, and by that same logic, after the first step, you can also feed out. So that first step, glucose 6-phosphate, can be involved in other different pathways. Does anyone have any other thing they can add? Yeah.
You're using a second equivalent of ATP, so as a result, you're investing in more energy, so you want to regulate that step.
Yeah.
Make sure you're not wasting any more energy.
Exactly. The same logic as the first, right? We're using ATP, which means that we don't want to waste extra energy. So all these are correct. The one I was looking for is the first step is not glycolysis specific. So if we phosphorylate glucose, that glucose 6-phosphate can go into the pentose phosphate pathway, or it can make glycogen. It can do a lot of things. This step is, once it occurs, this molecule is destined for glycolysis. So after this step is done, glycolysis will run to completion. So before we sacrifice a fructose 6 molecule into glycolysis, we want to make sure that our body is ready for it, and that's why we heavily regulate this step. And if that's not clear, it is on this slide. It's just the last bullet point on this slide. And then also, one thing that's important to note is I think this figure does a good job illustrating like the regulation of phosphofructokinase 1. Does anyone want to interpret this? Okay, great, because I would love to interpret it. So when we have high ATP, right, fructose 6-phosphate, we have more fructose 6-phosphate and less phosphofructokinase 1 activity, as we can see here. So if we have fructose 6-phosphate, we have low phosphofructokinase activity until you reach a point where you have so much fructose 6-phosphate that you might as well just send it down the pathway. But if you have very little ATP, you're going to have, at low levels of fructose 6-phosphate, you're going to have high PFK activity because you need to make ATP. So that's why this graph is shifted to the left. You need ATP, so PFK1 has to have a really high activity. That's why at low ATP values, this graph is shooting up at a low concentration, but at high ATP values, is staying low at this concentration. Are there any questions yet? Yeah.
When you say that the last bullet point that the second step is reversible, do you have to mean irreversible?
Sorry, so this is the third step, right? Our second step is the isomerase step right here, right?
Okay.
So this step is reversible. This third step is irreversible, which is why we regulate it pretty heavily. Yes.
When ATP binds like the active and the allosteric site, does it like choose the active over allosteric, which is why like at high ATP levels it can also bind to allosteric, or is it like equal?
It's equal, but if you have like a really high concentration of ATP, they'll bind to either. And like the more ATP you have, the more likely you are to inhibit the enzyme. If you have lower ATP values, it'll equally bind to either site, but you just have a less chance of like an ATP molecule bumping into this allosteric site or the active site. Any other questions? Okay. Step 4, which is, I'm going to skip this slide. So step 4, we have the splitting of a 6-carbon sugar into 2 3-carbon sugars. So the reactant is fructose 1,6-bisphosphate, same as the step before. And then we have dihydroxyacetone phosphate and glyceraldehyde 3-phosphate as our products. G3P is what makes the cut. DHAP does not make the cut. So we have to change that in the next step. The enzyme in step 4 is aldolase. So lase is something being cleaved. Aldolase, lyase enzyme, it's cutting things. It's important to note. And this is an endergonic reaction. Something to note that it does require energy. It is unfavorable. But it proceeds forward through constant and immediate removal of the products. So every 1 in 100 chance, this reaction will go through, and the products will be removed really quickly, making Le Chatelier's principle drive the reaction to the products. Anyone have any questions on that? That will become clear in later steps. And then reaction 5, our last reaction of the preparatory phase. This is actually my favorite reaction. So it forms 2 molecules of the same structure. So like I said, G3P makes the cut, DHAP doesn't. Here we're taking DHAP and turning it into G3P so it can actually go into the payoff phase. We have the enzyme triose phosphate isomerase. Here again we have an isomerase, which is the changing of isomeric forms. And then it can also be stand for as TIM. And this enzyme, which is like a fun fact, is the nearly perfect, it has nearly perfect catalytic efficiency, 2 times 10 to the power of 8. I think the perfect is like 1 times 10 to the power of 9. So the only limitation of this enzyme is the diffusion rate of the molecule. The molecule diffusing through the enzyme is the rate limiting step, not the actual conversion. So yeah, I have a friend named Tim, and that's how I remember this enzyme name. Any questions on the preparatory phase? Fantastic. Here is the payoff phase, which is steps 6 to 10. So every step from now on is doubled because we have 2 molecules. We have 2 molecules of G3P. So step 6, what happens? Right? Yeah. Okay. So the reactants G3P and NAD plus are used. And then the products is 1,3-bisphosphoglycerate, 1,3-BPG, and NADH. So this is actually the first energy-producing step in glycolysis because, as you all know, hopefully, NADH is used in the ETC to make ATP. And so this is a very big deal for the glycolytic reaction. We have the enzyme glyceraldehyde 3-phosphate dehydrogenase. So dehydrogenase is the name of an enzyme that's involved in redox reactions. So we transfer a hydrogen from a substrate to a coenzyme. And when you hear dehydrogenase, usually think NAD, NADP, or FAD. You'll see this name show up a lot in TCA. Electrons are removed from G3P and transferred onto NAD, as we can see. NAD plus goes to NADH. And it's an endergonic reaction, and it's reversible. So if it's endergonic and reversible, why on earth would glycolysis proceed? Well, because it's driven forward by step 7, which is highly exergonic, but I'll get there in the next slide. And one thing to note is in this glyceraldehyde 3-phosphate, we have a tiny molecule with an already huge negative phosphate group on it. In this reaction, we're throwing on another huge negative phosphate group, which just is like a recipe for disaster. We have two massive negative groups repelling each other on a tiny molecule. That just builds up a lot of energy, which is beneficial for the next step, which is this. We have reactants 1,3-BPG and ADP. So the product's 3-phosphoglycerate and ATP. That's our products. The enzyme is phosphoglycerate kinase. Like I said, kinase is phosphorylation. ATP, think kinase. And it transfers a phosphate from this high energy, negatively charged 1,3-BPG to ADP to make ATP. And so, like I said, step 6 was a little unfavorable. Steps 6 and 7 are coupled energetically and physically through substrate channeling. So they're coupled energetically, which means that net delta G is negative, but also the enzymes are like literally connected so that the products of step 6 go directly into the enzyme in step 7 so that this reaction proceeds. One thing to note, it's exergonic and it's irreversible. After this step, it cannot be reversed. So 1,3-BPG is even higher energy than ATP. That's why we can make ATP with this molecule. And then this step provides electrostatic relief from the two negatively charged phosphate groups on 1,3-BPG, like I said. Step 8 is the shifts. So it shifts phosphate to the center of carbon 2. So as we can see, we have a 3-phosphoglycerate with a phosphate on the third carbon, and we just shift this carbon to this phosphate group to the second carbon with phosphoglycerate mutase. Mutase is basically the same as isomerase. So yeah, when you think mutase, think isomerase, think changing isomeric forms. And so this mechanism forms 2,3-BPG as an intermediate. This is, I think, beyond the scope of the class. I actually didn't know this until I read this on this slide. But you form 2,3-BPG as an intermediate. And this note explains that. If you're curious, come up to me after, but that's not important for this exam, I don't think. Step 9, you lose an alcohol and you create a double bond to create the direct precursor to pyruvate. So as we can see, enolase, if you think of like an alkene, we have like that enolase here, and we're making a double bond. That's just a way to memorize the enzyme name. We're taking out a water, so it's a dehydration reaction, and we're making a double bond. So we're taking 2-phosphoglycerate, making phosphoenolpyruvate. And this is what leads to step 10. So we remove a phosphate to form a second ATP and pyruvate for the rest of cellular respiration. So the second ATP synthesis in glycolysis, this is our second ATP synthesis, along with step 7. And we have phosphoenolpyruvate and ADP. That makes pyruvate and ATP. The enzyme is pyruvate kinase. Kinase, I said a million times, is probably with ATP. And then this step is extremely exergonic because phosphoenolpyruvate has high energy repulsion due to the negative charge. So it's a tiny molecule with this negatively charged phosphate group and these negatively charged oxygens. So it kind of wants to get rid of that phosphate. And this step allows it to do that. And pyruvate can tautomerize to a more stable keto form. And it really wants to tautomerize to that keto form, which is another reason why it's driving this phosphate off and is favorable for the phosphorylation of ADP. That is glycolysis. Yeah. Any questions on that? Yes.
Wait, if step 7 is so exergonic and like irreversible, then why is there no bypass step for gluconeogenesis?
For number 7?
Yeah.
Isn't there a bypass step?
Not for step 6. Geez. Step 6, right? Glyceraldehyde.
No,
3.
step 7.
Step 7?
I'm pretty sure there's a bypass step. Am I wrong? Well, I didn't review that yet, so that's on me.
It's
Gluconeogenesis bypasses was 1, 3, 10.
Yeah, it is 1 through 10. That's what I was gonna say. The reason why it doesn't bypass 7 is because you can build up the products again from gluconeogenesis and it drives it the other direction. So that delta G that you see, it's standard conditions, but we know in the cell that's just varying concentrations. So with gluconeogenesis, you end up altering that ratio and it drives it towards the other direction. Because it's possible to have reversibility with these enzymes. That's why you don't have a bypass reaction.
The slides say exergonic irreversible.
Oh,
Okay, ignore that. It's not irreversible.
yeah,
It is reversible.
it is.
My mistake. Great question. Fantastic question. Any other great questions?
Also, I'll cover it in gluconeogenesis, don't worry. I'll clear everything up.
Okay, so then just to like almost wrap things up pretty quickly, we have 2 fermentation pathways. So the goal is to regenerate NAD because we need NAD in glycolysis to make NADH, but also to make glycolysis proceed. So if we have no reformation of NAD+, glycolysis will stop because we don't have a valuable step in the process. And so the way we regenerate NAD is with lactic acid fermentation or with alcohol fermentation. Lactic acid fermentation is pyruvate plus NADH, so we have pyruvate. And we make lactate and NAD+. So the enzyme is lactate dehydrogenase. Dehydrogenase, think NAD again. And so this is the reaction. One thing that's helpful for me is when you use NADH and go to NAD+, because one thing is getting reduced and one thing is getting oxidized, you're probably going to have one less bond to oxygen. So as we can see, pyruvate is becoming reduced to L-lactate because we have one less bond to oxygen. And so that's another way of thinking of oxidation. If that's confusing, come up and ask questions about that. Alcohol fermentation is we have pyruvate, which goes to acetaldehyde and CO2. The enzyme is pyruvate decarboxylase. And then we have acetaldehyde and NADH, goes to ethanol and NAD+, with alcohol dehydrogenase. So dehydrogenases, voice said NAD, NAD+. And so we have pyruvate with pyruvate decarboxylase. We lose the CO2 to get acetaldehyde. Acetaldehyde is then reduced to make ethanol with alcohol dehydrogenase. This is done in yeast, and then lactic acid fermentation is done in mussels and microorganisms. Any questions on that? Nice. Finishing off, we have the pentose phosphate pathway. This is so it's important to note NADPH is produced for fatty acid synthesis, and ribose 5-phosphate is produced for nucleotides. So NADPH, the red boxes, and ribose 5-phosphate, another red box. There's the oxidative phase, which is 3 steps to make ribulose 5-phosphate, which eventually makes, or 3 steps to make ribose 5-phosphate and 2 NADPH molecules. And then we also have the non-oxidative phase, which is a series of steps that recycles ribose 5-phosphate, or I guess ribulose 5-phosphate, into glucose 6-phosphate. So that turns it back to glucose 6-phosphate, and that's through a series of pathways. I think the key thing you want to note is that our pathways are connected. We have glucose 6-phosphate, which goes to glycolysis, but then it also goes to the pentose phosphate pathway, and then that can go to either form nucleotides, coenzymes, DNA, RNA, or it can go form fatty acids. So I think the key to this slide, while understanding the steps, is to note that everything is pretty connected in terms of pathways in the body. All right. Any questions? Okay.
All right, so I'm going to cover the next 2 lectures. So we're going to start with the TCA cycle. So let's just kind of give context as to where we are right now. So we went through glycolysis, we have a lot of pyruvate, and now we have to figure out what we want to do with it. So we have to start by converting our pyruvate molecules into acetyl-CoA before we can enter the TCA cycle. Okay, let's see.
Can you refresh the slides?
Yeah, I need to refresh that. Sorry, I made some like last minute
you're
edits. Okay, hang in there. I'm going to. Oh, okay. I guess it didn't show up on my end. That's fine. Oh wait, that's fine. Maybe I'll post like an updated version of the slides. I made some edits there, but in general, I guess we can talk a little bit about where we are right now. So the entire pathway in glycolysis, that happens in the cytoplasm. So, you know, basically like most cells can do this. However, the next part of the process, which is converting pyruvate to acetyl-CoA and then the TCA cycle, that's happening in the mitochondria. So just to give another recap of, you know, the organ with the mitochondria itself, it has an outer membrane and an inner membrane. That's going to be really important when we're talking about localizing our reactions. This is going to come into more play later on when we talk about oxidative phosphorylation. It's about the similar size as bacteria, so there is that whole endosymbiotic theory where, you know, the cell engulfed like another like bacteria and that ended up becoming the mitochondria. Yeah, I think that's the main points we need to talk about for that. But for the actual pyruvate dehydrogenase reaction, This is going to be happening in a complex called the PDH. So this is made up of 3 different subunits, so 3 different enzymes all working together, and there are 5 different cofactors. And so essentially the goal here is we have our 3-carbon pyruvate molecule. We basically want to kick off one of the carbons and add on our CoA group so that we end up with our 2-carbon acetyl-CoA group. And this is very exergonic, and so basically these reactions they're essentially irreversible. All right, so we're going to start off at subunit 1, so E1. And what we're going to start off with is decarboxylating our pyruvate, and then we're going to initiate the generation of acetyl-CoA. So what's going to happen first is pyruvate's going to attach itself near the enzyme complex, and then we're going to basically decarboxylate it and remove that CO2 molecule over here. And the other 2 carbons are then going to attach to TPP, which is a cofactor that involves thiamine, which is vitamin B1. I think that's beyond the scope of the exam, but that's just extra information. What happens after that is the TPP with our 2 carbons, it's going to transfer those carbons to lipoic acid, which is another cofactor. So this has like this arm that like swings around, and that's just what makes the reaction super coordinated. And so now when we get to the lipoic acid with our carbons, it is now it should be reduced, it's reduced and it's acetylated, so we have our 2-carbon group here. So we transition from this oxidized version to the reduced version, so we have the extra hydrogens, and then we have one of the sulfurs of the lipoic acid connected to our 2-carbon group. So now we're in E2 of the PDH complex, and this is where we're actually going to form acetyl-CoA. So our lipoic acid is going to transfer the acetyl group to our free coenzyme A, and so attaches at that thiol group. And now, here it is where the lipoic acid is reduced. And so essentially here at this point in the PDH complex, our acetyl-CoA is basically formed. But what's really important is that this process has to keep repeating itself in order to keep making more acetyl-CoA for our TCA cycle. So what we have to do now in E3 of the PDH complex is to reset. So we have to reoxidize our lipoic acid back to its original form, and that's how we can make more of our acetyl-CoA. And that's going to be done through FAD. So as lipoic acid is being reoxidized, FAD is going to be reduced to FADH2. And so this is going to seem familiar later on as we go through the different steps of the TCA cycle, a lot of oxidations, a lot of reductions. And then to reset the FADH2 back into FAD, we will have our NAD+, which is another cofactor. And so as FADH2 is being reoxidized back into FAD, the opposite happens for NAD+, and that's going to be reduced to NADH. And so essentially with all of these different cofactors, I think once again with in terms of structures for the purposes of this exam, I think the structures are all really big and complicated, so you don't have to draw it out by hand, but definitely be able to recognize the key parts of each cofactor that's important for the synthesis of these new molecules. And just be able to recognize them on the exam because you may be asked to name them. And so with the PDH complex, this is a really important transitory step in order to bridge glycolysis and the citric acid cycle. And so this will require some regulation. So the PDH complex, it can be inhibited by a lot of things such as NADH, acetyl-CoA, ATP. And that makes sense because if we have a lot of acetyl-CoA, we don't need this process to keep occurring again. And so that's why we're going to have product inhibition, and it's going to stop the PDH complex from continuing to do its work. On the other hand, if we don't have enough acetyl-CoA, some molecules like NAD+, AMP, ADP, and free CoA, these can help upregulate the synthesis of acetyl-CoA and just drive more production of it through the PDH complex. Another thing is that we can do some modifications with enzymes. Oh, sorry, is that a question?
Yeah, it is. So is the ATP and NADH that's being negatively regulated, is that relating to the TCA cycle, or is it just between pH?
I think it's coming back from the TCA cycle because we're not generating ATP and we're not really, the NADH is also coming from the TCA cycle later, so I think it's all just coming back. Because, so that wasn't mentioned, but I think all of these molecules that are inhibiting, a lot of them are coming, they're coming back up because if we have the TCA cycle running a lot, then we don't need to keep making more acetyl-CoA, and that's why the product inhibition is going to come back. So I guess what you'll notice from a lot of these regulation pathways is that it doesn't, when we have product inhibition, it's not necessarily like an immediate inhibition back to like the previous step. You can have molecules from all the way downstream of these pathways go back up to the very top to also inhibit this process. Yeah.
Also with PDH, we know that it's going to convert it to acetyl-CoA, right? And we know that acetyl-CoA will be the carbons that come out of the TCA cycle as carbon dioxide. So what does this mean, and what am I trying to get at? Is that acetyl-CoA is not gluconeogenic, which means acetyl-CoA cannot be used for gluconeogenesis to form glucose. So that's why we heavily regulate this step, because if we form that acetyl-CoA, it's just going to go through TCA. We can't use it for anything else. And you guys will learn later on about fatty acid synthesis, but that can't be converted to glucose itself. But there's a whole thing on it. So as of right now, all you need to know is that acetyl-CoA is going to be used up in Krebs cycle, and you can't use it for anything else for this exam.
Yeah. Any other questions? Okay, sounds good. So just one final note on regulating the PDH complex. It can also be modified by additional enzymes, mainly PDH kinase and PDH phosphatase. So here's the one thing that I really want to emphasize when talking about this is that normally in biochemistry, a lot of the phosphorylation, it tends to activate certain molecules, and removing that phosphate group tends to inhibit them. In this case, it's the opposite. So PDH kinase, when it's adding that phosphate group to the PDH complex, it isn't activating the E1 subunit. And conversely, when we take away that phosphate group through PDH phosphatase, it activates E1. All right, so now we have our acetyl-CoA molecule generated. Now can actually go through the TCA cycle. And the purpose of this is to oxidate all of our products from glycolysis, and it's going to generate our cofactors and some ATP. It's a very minimal amount, but the key thing is generating the cofactors that will then be used in the electron transport chain. So just to give a very, just macro scale like understanding of what the TCA cycle does, for each acetyl-CoA that's going through this cycle, we end up generating 3 molecules of NADH, 1 molecule of FADH2, 2 molecules of CO2, and 1 molecule of GTP/ATP. And this is all happening in the mitochondrial matrix, so this is past the second membrane, is just within that matrix space. All right, so we're starting off with a condensation reaction. So it uses Claisen condensation. You may or may not remember that from Orgo 2. But essentially in this first step, the acetyl-CoA that was just generated, it's going to be combining with oxaloacetate, and this is the molecule that's made in the very last step of the TCA cycle. And so it's basically bringing, the end of the cycle back to the beginning. And they're going to combine together into a molecule called citrate. And I guess the enzyme is called citrate synthase. And so a lot of the enzymes that you'll see in these reactions, they're all named, I think, in a very obvious way, so it makes sense that citrate synthase ends up making citrate, for example. Yeah, so this is just the TLDR. We have our 2-carbon acetyl-CoA. It's combining with a 4-carbon oxaloacetate, and then we have a 6-carbon citrate molecule. And here we also add water, and that coenzyme A group is being released. All right, so after citrate formation, then we're going to isomerize it. We're going to make isocitrate. So this basically takes like 2 sub-reactions, and they both use the enzyme aconitase. So in the first part, let's say reaction 2A, a water molecule is being removed, and so it dehydrates the citrate into a molecule called cis-aconitate. So if we see here, we have our hydroxyl group here that's being removed, and so it generates a double bond here in the cis-aconitate. What happens then in the next part of the step is that water is being re-added, so a rehydration reaction, and all it does is it's going to attack our double bond, and it's going to add that hydroxyl group on a different carbon, and that's all it is. It's just basically moving that hydroxyl group from one carbon to like the next one. Yeah, so that's just another TLDR step. And so once isocitrate is formed, then we are going to do oxidative decarboxylation. So yeah, we're going to lose a carbon, and then through that, we're going to form an NADH. So in this step, the isocitrate is going to undergo a reaction with the enzyme isocitrate dehydrogenase, And this is where we have our NAD plus as a cofactor. So as the isocitrate's being oxidized, our NAD plus is being reduced into NADH. So this is where our first NADH molecule is being generated, and that's where our energy is being stored. And then in the next part, so isocitrate's being converted into alpha-ketoglutarate, and what happens then is it's going to basically react with the alpha-ketoglutarate dehydrogenase complex, and this is where our second NADH is being formed, so this is happening in 2 successive steps. Our sugar is being oxidized, and now we have another cofactor being generated, and that results in succinyl-CoA, and this is a very exergonic reaction. We can see that delta G here, it's like minus 33.5 kilojoules per mole. So yeah, very, very exergonic, basically irreversible. All right, so now we have our succinyl-CoA, and this is where we have our substrate level phosphorylation, and this is where our 1 molecule of GTP/ATP is being formed. So our succinyl-CoA is going to react with succinyl-CoA synthetase, and is also going to use a GDP plus an extra phosphate group as a cofactor. So as succinyl-CoA is being converted to succinate, we have our CoA group once again being removed, and we have our GDP plus the extra phosphate group that's being combined to form our GTP. So that gives us just a little bit of energy, but it's definitely not enough, so we're going to have to go through additional steps later in order to get as much ATP as possible. All right, and now in the last 3 steps of the TCA cycle, we're essentially just doing an alkyl chain oxidation. So we're just converting our succinate into oxaloacetate, and that's just going to bring us back to the beginning of the cycle. So we're going to have to introduce a double bond first, and that's going to help us create our FADH2 cofactor. We're going to add a water molecule to add a hydroxyl group, and then we're going to oxidize it to form that carbonyl that you see in oxaloacetate over here. So in step 6, we have succinate reacting with succinate dehydrogenase and FAD. So succinate's being oxidized, FAD is being reduced to FADH2, and that's going to give us our fumarate. So this is going to be a trans double bond. Not very exergonic, basically 0 kilojoules per mole. And now we have more of our energy stored in an additional cofactor. And keep in mind now that all of these steps here don't involve, the addition or loss of carbon. These molecules now all are 4 carbon molecules, and that's the same as oxaloacetate. And now once we have fumarate, what we want to do is break that double bond, and we want to add our hydroxyl group. So fumarate is going to undergo a reaction with fumarase, and that's going to give us L-malate. And this is very important because only the L isomer of malate is going to work. If the questions on the exam trick you by asking about like D-malate or something, just know that it's not going to do anything. We can only use L-malate. And so once it's converted to that, then we can prepare for our oxidation step in order to generate oxaloacetate. So in our last step, we're gonna oxidize L-malate into oxaloacetate, and that's going to help us generate our last NADH molecule, because the oxidation of L-malate will result in a reduction in NAD+. And so once we have our oxaloacetate molecule, we're basically at the end of the TCA cycle, and we can basically rerun it again to generate more cofactors for for ATP synthesis later on. All right, so once again, as with a lot of these metabolic pathways, there is a lot of regulation in the TCA. And so I would say the key ones to be on the lookout for are the initial step, where we're combining our acetyl-CoA with oxaloacetate into citrate. And then also here, when we're converting isocitrate to alpha-ketoglutarate, and also alpha-ketoglutarate to succinyl-CoA. So these are the main steps that are being heavily regulated. These steps are also the ones that are very exergonic, so it makes sense that these are the ones that are being controlled by with a lot of these different other molecules. And yeah, and this is just a summary of TCA cycle regulation in table form. So you can go back to this slide afterwards. We're going to move on for now. And so one thing that's really important to understand about the TCA cycle is that it's carbon neutral. And so I think Alex has already mentioned this a little bit beforehand, but acetyl-CoA, its only purpose here right now for the purposes of this exam and for these lectures is that it's just going to undergo the TCA cycle to generate cofactors in order for us to get more ATP. But the thing is, acetyl-CoA by itself, it cannot replenish the TCA cycle intermediate. So if we're running low on oxaloacetate, for example, we can't run an acetyl-CoA molecule through the cycle just to make more oxaloacetate and have it as like and have it on standby. It doesn't work like that. So what we have to do is we have to use some anaplerotic reactions. So it comes from the Greek for like to fill up, and essentially what it does is these reactions are there to replenish our TCA cycle intermediates as needed if we are running low on them. So this table here shows 4 very common ones. I think the most common one would be this top one here, where we have our pyruvate undergoing a reaction with pyruvate carboxylase, and that's going to form oxaloacetate. And you're going to notice that a lot of these anaplerotic reactions, they end up forming oxaloacetate as an end product, and that's because normally in the cell we have very low concentrations of oxaloacetate, and so if we need it for any reason to generate a lot of ATP, this is when those anaplerotic reactions would occur. And also it's important to note that these anaplerotic reactions, some of these, they're specified to specific tissues or organisms. So even though a lot of these generate things like oxaloacetate as like an end product, it doesn't mean they're all happening at the same location. So each reaction is like localized to different areas of your cell or body. And lastly, I just want to touch a little bit on the glyoxylate cycle. So this is not something that happens in vertebrates. This happens more like plants, invertebrates. But this allows for the conversion of our 2-carbon acetyl-CoA molecule directly to 4-carbon molecules. And this is also why vertebrates, so like us, we cannot survive on a primarily fat diet. So this reaction, it takes place in the glyoxysome. And what happens is succinate is brought into the TCA from our mitochondria. And so a lot of these reactions at the beginning, they're very similar to the TCA cycle that we see in humans. However, there are 2 unique enzymes here. The first one is isocitrate lyase. So we have our isocitrate molecule here, similar to, from the TCA cycle. What happens is it basically chops that isocitrate molecule. It forms succinate, and the other part of it forms glyoxylate. And this glyoxylate molecule is going to then combine with acetyl-CoA, and that's going to be joined together with an enzyme called malate synthase. That's going to give us malate, and then the reaction, same again as the TCA cycle, it brings us back to oxaloacetate. So this is kind of like a shorter condensed version of the TCA cycle, but we can't do this. And so yeah, the net reactions here, we have 2 acetyl-CoA molecules plus NAD plus 2 water molecules, and that gives us succinate, 2-CoA, an NADH, and an H plus. Yeah. Yes?
Is this an anaplerotic reaction in plants? So can they use the succinate from there to replenish?
Yes, exactly.
Yeah.
So plants have the ability to use the acetyl-CoAs. We, as vertebrates, we lack the, what is it called again? I can't remember. But there's a specific one that allows it to form succinate. So yeah.
Yeah, so the main thing is, you're basically right, because we have this free acetyl-CoA molecule. It's being combined with these other intermediates in order to form malate, which is an intermediate in the TCA cycle, but we can't do that. A lot of times, I think in the exam, they like to trick you by asking you about like the glyoxylate cycle in humans, or like, something to do with converting acetyl-CoA to like other molecules. Don't fall for that. We can't do that in humans. Does anyone have any other questions? All right. Okay, so now we're going to talk a little bit about oxidative phosphorylation. So this is, where all of our cofactors are being generated, and now we can actually use them to generate the bulk of our ATP in order to get as much energy as we can. So continuing off from where we left off with the citric acid cycle, we have our 1 molecule of ATP, we have our 3 NADH molecules, and we have our FADH2. But now we need to do something with them, and this is where it happens. Okay. Yeah, I think the slides are a little bit out of order. But yeah, so now the electron transport chain and the oxidative phosphorylation, that's happening along the inner membrane of the mitochondria. So you'll see that a lot of the action is happening either, very close to the matrix, on the membrane, or just outside the intramembrane space. But we're going to cover that in our subsequent slides. Yeah, so in the electron transport chain, we're going to see a lot of different complexes, a lot of cofactors. I think, once again, just be aware of, the function of these. You don't need to draw any of them once again. One example is, oh yes.
Could you go back to the slide before?
Mm-hmm.
Yeah, so, intuitively, what helps remember, like, that NADH is being pulled from those, or, like, is it, like, for example, what helped with malate to oxalic acetate was that it's oxidation, so NADH had to get reduced? Like, what helps you, like, intuitively know which steps have the electron carriers?
They typically will be called dehydrogenase. So, like, for example, citrate synthase, there's no oxidation reduction. Aconitase doesn't have that dehydrogenase, so there's nothing. There's alpha-ketoglutarate dehydrogenase, that one does, and you see it has the NADH. Then there's succinyl-CoA synthetase. No, not synthetase. Sorry. I thought the first one was isocitrate dehydrogenase. Sorry. Then the next one is alpha-ketoglutarate dehydrogenase, and then succinyl-CoA synthetase is going to have the GTP formation. That one doesn't have dehydrogenase, so you're not necessarily reducing anything. Then succinate fumarate is succinate dehydrogenase. So like it's in the name of the enzyme. If you ever see dehydrogenase or oxidoreductase, you can assume NADH cofactors are involved. That's like a really good general rule of thumb.
What's the rule of thumb for ATP?
Synthetase or synthase. Synthase is very like I would be cautious about it, but synthetase because it has the taste or in it. That one has ATP or GTP. You just have to remember which one is which.
Okay.
Yeah. Does that help?
Yeah, thank you.
Yeah. I think just one note is that in the nomenclature, you'll sometimes see enzymes called like synthetase or synthase. I think back in the day, there were like slightly different definitions, but for the purposes of like now in this class, you can just essentially treat them as like the same thing. Yeah. Just want to bring that up. Yeah. Yeah, that's a good question. So I think the main thing is just it depends on which organism you're in. So I think for humans, we make GTP, and other organisms, they make ATP. But once again, they're essentially interchangeable in this case. It's just one molecule of it that just generates a little bit of energy.
That's why I wrote this on the board. Like, I know it's probably a terrible drawing, but we do end up putting that GTP with this first reaction with the succinyl-CoA going to succinate. But there's this other reaction that they don't show you, which is like phosphorylating the ATP. The difference between them is just that obviously they have a different nucleobase. One has a guanine, the other one has an adenine. That's just a difference, but they're interchangeable because they can exchange the phosphate groups.
Yeah. All right. Any other questions?
Yeah.
Could you re-explain the thing you said from the last slide about how like humans can't use acetyl-CoA in the same way that plants can?
Yeah.
Do you want me to take it?
Yeah, sure.
Yeah. So this glyoxysome is a specific type of organelle found only in plants, or better yet, invertebrates. And so if you have this glyoxysome, you're going to have this enzyme called isocitrate lyase, which is an alternative pathway. Remember, in TCA, we have isocitrate and it goes straight to alpha-ketoglutarate in normal Krebs cycle. But this has an extra enzyme that offers that alternative pathway. And it's exactly this alternative pathway that you can combine another acetyl-CoA to form the succinate. And that replenishes the TCA cycle. Does that make sense? I can also draw it on the board with the two pathways coming in, and I can explain it a little bit. To save time. Yeah, I'll just draw it.
Yeah, go for it.
You can keep going, that's fine. I'm going to take a little bit.
Okay, sounds good. Sounds good. All right, so yeah, I was just talking a little bit about, so these are some iron sulfur clusters. You'll see them in some of the complexes. I think in general, when we're going through, the oxidative phosphorylation, I don't think you need to know like the nitty gritty of like each individual like sub-reaction that happens in each complex, but just know that iron sulfur clusters are here. And the main point of this is just, it just increases like stability of everything. Iron is very good at that, and it's also being bonded to like our sulfurs, so from like the cysteines. Yeah, and it also changes oxidation state. So when we're talking about moving electrons from one molecule and jumping to another one, that iron sulfur cluster also helps with that. Some other molecules that you should also be aware of, once again, you don't need to like draw it, but definitely be able to recognize it. One is like ubiquinone. So this one comes up quite a lot, as we'll see later. So here at the top, we have ubiquinone. We also abbreviate it just as Q. This is a fully oxidized version of it. As we can see here, we have our carbonyl. And then we can also have it as like a semi-quinone radical. Yeah, so basically we're adding on one electron, and then if we add on another one, then we have a fully reduced ubiquinol, and that we abbreviate as QH2. So you might see me use like the abbreviations or the full name interchangeably. Another class of molecules that you'll see a lot in oxidative phosphorylation are cytochromes. And so, yeah, heme is a prosthetic group of that, and you know, we might recall that from our previous lectures from like 1 to like 6. Yeah. Okay, so I'm not going to touch too much on this slide, but just, you know, be aware of electrochemical potentials in terms of, you know, like redox reactions again. I think Ryan covered that pretty well, so I'm not going to redo that, but I think just have a general grasp of, you know, the reduction potentials and everything of these half reactions, and that kind of explains the flow of electrons from, you know, complex 1 to complex 4, for example. Okay, so this diagram does a pretty good job of giving an overview of the electron transport chain. So we have complexes 1, 2, 3, 4, and then we have ATP synthase at the very end. I think for studying purposes, I would recommend having like a blank version of this and trying your best to kind of draw it out step by step and, you know, see how that flow of electrons and protons are happening, where that's occurring, and how that drives ATP synthesis later on. Yeah, I would say that's a pretty useful strategy, and also I think by doing this it gives you a more visual reminder in case people here are more visual learners. If you don't like writing all like the reactions like in a line, I think this can be a useful strategy. Yeah.
You want to explain real quick?
Yeah, we're gonna take a segue.
I know this is like a very poorly drawn diagram. I wish I could like use my iPad, but essentially this is the TCA cycle, right? It's circular. It keeps. We know what it does. But glyoxylate, this is now we're talking about plants. It has both these pathways going simultaneously. And what I was saying is from isocitrate to alpha ketoglutarate in citrate, there's that one potential pathway. But there's alternatively that other one where it goes from isocitrate to glyoxylate plus succinate. And it's exactly the succinate that will replenish this intermediate. Does that make sense? So both of them are happening simultaneously in plants, and that's exactly what allows them to survive on acetyl-CoA, because acetyl-CoA can now form these endopleurotic reactions. And if you remember, there's a certain way it has to do with I think like malic enzyme or something like that. You'll learn about that later. Don't worry about it right now. But acetyl-CoA can replenish in plants. Does that make sense? Yeah, you want to take a picture? That's fine.
All right, we good? Okay, sounds good. So we're at the beginning of our electron transport chain. We're going to start off with complex 1. It's also called NADH ubiquinone oxoreductase. So that gives a pretty good idea of, you know, what this complex does. We have our NADH going in, and through a series of reactions, we're going to have 4 protons being pumped out, and we're going to have another 2 protons from our NADH. That's going to be transferred to ubiquinone. So this is the fully oxidized version, so Q. Yeah, the key points here, NADH gets oxidized, ubiquinone is reduced. Yeah, this complex only uses like NADH. So just be aware that when we're talking about the electron transport chain, even though we have complexes like 1, 2, 3, 4, in reality, because we have 2 different electron carriers, we have NADH and FADH2, the sequence is actually more like 1, 3, 4 and 2, 3, 4. So just be very aware of that. There's that localization between NADH and FADH2. All right, so we have NADH doing its thing, and so now let's look at what FADH2 does when we're talking about the electron transport chain. So it's going to go directly to complex 2, also called succinate dehydrogenase, so we have that connection from the TCA cycle. And so what we're doing here is that FADH2 bypasses complex 1, it comes over here. Yeah, and so basically through the iron-sulfur clusters, we're also going to be transferring our electrons from FADH2 to ubiquinone. So it's just 2 different ways of doing it. It's just that one complex uses 1 electron carrier, the other one uses a different one. That's essentially what the difference is. And so now our electron carriers, we don't really need them anymore at this point because all the electrons and the energy they have, that's all been transferred to our ubiquinone. And so now we're at complex 3. So this is like this gigantic protein with a bunch of different components. So we have our heme groups, we have our iron-sulfur proteins, we have cytochromes. And so here at complex 3, our ubiquinone is going to basically transfer the electrons to cytochrome C. And so if you see the mouse here at the very top, cytochrome C is like the one component here in this process that's not embedded within the inner membrane of the mitochondria. It's kind of like free floating throughout the intermembrane space, and that helps it, move the electrons from complex 3 to complex 4, which we'll see later. The only problem here right now is that ubiquinone carries 2 electrons at a time because we have QH2, but cytochrome C only carries 1 electron. So what do we have to do? We have to do a 2-step process in order to transfer all of the electrons over, and that's what we're going to call the Q cycle. So what happens is we have 1 QH2, so this is the fully reduced version, so ubiquinol. It's going to give 1 electron to cytochrome C, and that's its full capacity. And it's going to pass off 1 electron to an MTQ, and that creates a like a radical version of it because it only has 1 electron instead of 2. Then what happens is we have another QH2 molecule come in. It's going to give one of the electrons to cytochrome C, and another one to that radical Q, and that's going to basically reform QH2. So through this like 2-step process, sorry about that, we're going to have our cytochrome C's fully loaded, and we're going to remake our QH2 so that this transfer can keep happening again without generating like a bunch of radicals in the process. Yeah. All right, so now we have our cytochrome C's. They're free floating around the intermembrane space. Now we're going to take them to complex 4, which is also called cytochrome oxidase. And this is where that final oxidative reductase step happens. So when the electrons move from cytochrome C to complex 4, the electrons are going to be stored at a iron copper center, and then it's going to wait till once the electrons arrive, then it's going to pump out 4 protons into the intermembrane space, and it's also going to form 2 molecules of water. And so here, by this point, we're going to have a pretty steep proton gradient. So a lot of the protons are being pumped from the matrix of the mitochondria outside into the intermembrane space. So on the outside, we have a bunch of protons. On the inside, we don't have a lot. And this is going to be really important, like in the next step, when we're actually going to be creating our ATP from the ATP synthase. So here, I also just want to note that NADH is much more efficient at generating like ATP than FADH2. So if we look here, like patterns of 8 and 12, and then FADH, it's a pattern of 4 and 8. And now with our very steep proton gradient and such, we can actually use our ATP synthase protein to make the ATP that we've been wanting to do for this entire time, all the way from glycolysis till now. This is where we're going to be making the bulk of our energy. So I'm just going to briefly walk over the structure of ATP synthase. So there is the F0 subunit and F1 unit. At the top here, we have our alpha, beta, and gamma subunits. So the alpha and beta subunits, they're kind of like alternating and kind of forms like a wheel. We have our gamma subunit here. It's kind of like the shaft of like a rotor. And then we're going to have ATP being bound tightly up here. And then at the bottom, we have our F0 subunit, and that's going to be made up of, I guess we can call that like, we have our F0 unit. That's going to be like the rotor. That's going to be spinning everything around. And also, I just want to note that this diagram here, it's kind of upside down depending on which way you want to look at it, because this whole time when we look at the electron transport chain, we have the matrix of the mitochondria at the bottom of the page. We have our intermembrane space at the top. In this diagram, it's being flipped, so the matrix is at the top and the intermembrane space is at the bottom. But this just gives a better idea of, like just how the ATP synthase works and like just like the rotation structure. All right, and so the energy requiring step when we're using this protein is the removal of ATP itself from the subunits. So if we look here at this diagram, I want to focus more on the right side here. So we have 6 individual subunits, so they're alternating between alpha and beta. And at any time the ATP synthase protein is working, there's basically 3 different configurations. So we have one configuration here where there's nothing in these subunits, the ATP is just left. We have another configuration here where we have ADP plus our phosphate group, and that's waiting to be combined into ATP. And then our last configuration is that we have our fully formed ATP molecule, and it's just going to be waiting to be released. Yeah, and so the gamma subunit, it's going to be what turns, the ATP synthase wheel, and that's what's going to generate our molecules. And then the protons, that proton gradient from, these 4 complexes, that proton gradient is going to turn a glutamate helix, and that's also going to propel that rotor in order for ATP synthase to work. Yeah, so once again, this is just another representation of those of the F1 subunit with all the different with all of the different configurations. So I think this year they added more content where they talked about additional experiments that were used to validate how ATP synthase worked. I think I can't speak for what's going to show up on the exam. I think the best thing to do is not focus on the nitty-gritty of each individual experiment, like the methods they use, like each individual step, but more so just look at the bigger picture and see how this experiment validated one aspect of it. So these subunits have to work together. You can't just isolate one of them and expect ATP synthesis to happen. So yeah, I would say focus on that versus the precise details of the experiments. Does that make sense? Or does anyone have concerns or questions? Okay. Sounds good. So we have our ATP that's being made, so congrats. We have a lot of our energy that we have now. But now we have to talk about ATP transport. So when we make our ATP molecule, we need some of our components back. Like we need ADP again, and we also need phosphate groups. Otherwise, this process cannot continue to work. So what happens is we need two transporters. So the first one is an adenine nucleotide translocase. This is an antiporter molecule, and so all that means is we have our ATP. It's being moved from the matrix to the intramembrane space, and in the opposite direction, we're having an ADP that's moving into the matrix, and that allows and that's going to be added to our ATP synthase, and that's going to generate more ATP later on. The other transporter that's also important in this case is a phosphate translocase. This is a symporter. So what happens is we have our phosphate group here. It's going to be moving into the matrix, but along with that, we're going to have a proton moving together with it. And this is important because our phosphate group is negatively charged. We have our proton that's positively charged, and so this is going to make the process electrically neutral. And so these two transporters, they just allow for the balance between removing ATP versus bringing back the components needed to generate ATP back into the mitochondrial matrix so that it can be used in ATP synthase. All right, and now I just want to touch a little bit, it's a little bit of a segue, but talk a little bit about some other transporting shuttles. So one important thing is that NADH and slash NAD+, that doesn't go into the mitochondria by itself. It has to be converted. So what we have here is this aspartate malate shuttle. And so essentially what happens is we have our NADH here, and that's going to help convert it to malate. And then the malate, it's going to be what's going through the transporters. And once it's inside, we can then do some more reactions there, reconvert it back to oxaloacetate. Then through some aminotransferases, that's going to give us aspartate, and then it can move back and forth. So essentially what you need to know is that malate and aspartate are the molecules that are going through these transporters. We can't just have NADH by itself. And then the other one is the glycerol 3-phosphate shuttle. This mainly happens in muscles. So what happens here is our NADH is being oxidized back into NAD+, and we have our DHAP, which is from glycolysis. That's going to be converted to G3P, so glycerol 3-phosphate. And so once it's in the glycerol 3-phosphate form, it can then basically be transported. And in that process, we have our FAD being reduced to FADH2, and that's going to help, and that's going to be at complex 2. And then when we transfer electrons then to ubiquinone to generate QH2, then we can proceed again with complex 3. And this is really important because this previous malate aspartate shuttle, it's pretty slow. The glycerol 3-phosphate one's a lot faster. And here you can see why it's being used in muscles because, let's say you're in a situation where you need a lot of energy really quickly in order to move your body for something, and you need ATP fast. This is when that shuttle is really important in order for you to basically convert the NAD plus an NADH and transport it in, and then basically undergo the electron transport chain. All right, and one other thing is that we have our electron transport chain, so we're going through the complexes, and then we're using ATP synthase to generate ATP. What happens is sometimes this process can be uncoupled, which means that once we go through all the different complexes, ATP synthase doesn't work for whatever reason. It could be a mutation in the enzyme. It could be some sort of chemical reagent. You might be asked this on an exam. I think my exam last year actually had a question. Sorry. My exam last year had a question on this where there was like a chemical mutagen that basically uncoupled the electron transport chain and the ATP synthase and asked like what would happen to it. But in general, just know that this happens. And what happens is if we can't generate our ATP, all of that energy is just going to be dissipated as heat. And sometimes this can be really important. So, you know, newborn babies, they need a lot of this heat. And that's a really easy way to get that extra heat. This is also, the heat generation, it's mainly found in brown tissue, so brown fat versus white fat. And we also have animals that are adapted to the cold. A lot of them will have this brown tissue with the uncoupling, and that allows them to generate more heat in order to survive in cold temperatures. Yeah, so essentially what happens is we have our hydrogen flowing along the gradient, and then heat's being generated and dissipated throughout the body. All right, and lastly, I just want to talk a little bit more about regulating ATP production at the step. So the main thing is that ATP synthase itself can be inhibited by being cross-linked together. And so this can be done with an inhibitor molecule called IF1. So that links these two ATP synthase molecules together and it kind of locks them in place, and so it can't do its usual job. And then besides that, just inhibiting any step in like glycolysis or the TCA cycle, that usually does the bulk of the regulation, because by this point, if we're in the electron transport chain, most of the time you're assuming that you just want your ATP, so there's not as much regulation there, but sometimes, you know, you have these inhibitors there. You can also have, once again, mutagens, mutations, things that, you know, can stop like ATP generation from happening. But usually the bulk of this is going to be happening in the previous steps, as we mentioned in our previous lectures. Question?
I'm a little confused. So that's a good thing or bad thing if it's uncoupled and heat is dissipated?
I think
Yeah.
there is some advantage for uncoupling. So there is a reason why our thing is only 30% efficient. That's because we have to keep a constant internal heat energy. So like for mammals, we want to stay warm, right? So that's why we have this uncoupling. And sometimes we get cold. So I don't know if this is true or not, but I'm just like speculating, is that you can secrete like some molecule that will uncouple it, almost act like it's an uncoupler. That could be an example. And perhaps you'll see maybe more efficient ATP synthase in the cold-blooded animals. This is just like all like conjecturing, but hopefully that makes sense in terms of physiologically why we have this in the first place.
Yeah. I think just to add on to that, if you have questions on the exam where they talk about, you know, like a scenario like this and it requires like applications, really think about the context as to like how this is happening, because that's going to help you determine whether or not the situation is like actually good or not. Yeah, because I think with most things like biology, like I guess you can't look at a situation like in isolation. You have to think about the wider context as to why this is happening. So, you know, one thing happening, it might be good in one context, but it might be bad in a different context. So you just want to be really aware of that when you're answering these application questions. So, you know, always think about the bigger picture before diving into your answer, because that will usually guide you towards the correct answer and what we want to see. But yeah, otherwise, are there any questions about these last two lectures at all? Okay, good.
Do you want to take a 5-minute break? It's fine if you guys don't want to, but if you do, it's all good. And you guys can just come up and ask questions too for the 5 minutes. Up to y'all.
5-minute break? Okay.
Okay, we'll do 5 minutes.
Be ready. Be ready. Be ready. Ain't nobody gonna ask
Hi, what's up?
Nobody gonna ask questions. So
I feel bad for people that leave.
you gotta tell them my favorite acronym. The TCA.
How you doing? I know. That's how it usually is.
Yeah, I think think that's a good amount of detail. I think, you know, have a complex
Can I please?
Yeah, I think
No, I know.
That was my favorite. You gotta say this. You gotta say this. You gotta say that one.
yes,
That's right.
yes.
That was like
All
my
the
biggest
protons that form in the
in the
intramembrane.
intramembrane space,
Yeah.
which is between the outer membrane and the inner membrane,
Yes.
separated
Yes.
from the matrix.
Yes, because this is on the inner membrane. Yeah, and then yeah,
That's so
the electron movement. Yeah, that looks good. I think. Yeah,
Okay.
Well, that's sufficient. Yeah.
But then, like, if you burn up all your fat, so that's why this one, you actually. Okay, we don't need that complete supplement to that. Okay, I think that's how it was. Don't ask me.
Okay, so your question is, you have protons on like the inside here that yeah. Okay, that's a good question. Okay. Okay, I want to say that no, we have our protons being pumped out. Right. But also at the same time, we have the protons and like the electrons that are being transferred from those electron pairs throughout the complexes. And so that's not going to go outside, that's all within this like membrane and on the inside, because we can see here that these complexes are being embedded into the membrane, and also it's going to be partially like in the matrix itself. So I guess that's I'm not sure that's like the best answer to your question, but because like a lot of these are like inside, I guess that's just why you know some of the protons and electrons will stay inside. Like it's not all of it's going to go into the engineer. Yeah. Do you have other questions?
Mine is, it's okay if you don't know the answer. Yeah. I'm having a hard time like remembering this compound because I can't figure out like mechanistically how to get from here to here. And I was wondering if you maybe have notes. I also couldn't find it like in the slides. And the rest of them I figure out, and that was helpful. So much.
My best guess, which I cannot confirm whether it's right or not, is that so we know that that's all chopped off. I think what's happening is because this enzyme is basically chopped off I think what's happening is once that enzyme is happening, I want to see what's happening is like there's going to be like tremendous erosion of that's going to be happening. So that's my best guess and if it's happening. Yeah, I can't confirm that, but yeah, knowing that this enzyme is top of and that's how you get really like your CH, like carbon hydrogen phosphate. Yeah, no problem. Yeah. So I think you definitely need to know the enzymes for like the main pathways, so like glycolysis, PDH, TCA. I think the smaller pathways, I doubt they're going to ask what the nitty-gritty about, like, oh, like what enzyme transfer helps convert, you know, something to something in this shuttle. I don't think you need to worry too much about that. For those smaller pathways, mainly just understand why they're there. Yeah, focus the actual like memorization and structure of like the bigger pathways, because that's what's more important.
The general structures for like all of the
I would say like pH complex, TC, I think those are pretty fair game to structure recognition. I think gluconeogenesis is less. I think maybe but less. I remember last time they were testing the main like structure recognition was all like yeah, I don't want to speak for the professors in the exam because they very well may have changed it, but Yeah, I think recognizing the structures can be important because then when you see like, okay, the substance looks like this and the product looks like this, you can kind of then figure out like what exactly happened. Like, was a hydroxyl group lost, was a phosphate group added. That can also help you remember like, okay, what are the enzymes being used in that step, because a lot of times they're being named after what. That's my best guess. But no problem. Yeah, it's good.
Yeah, can you explain a little more about like the two transporters and like the? I was kind of confused about that.
The two transporters.
Like how the two transporters, because it can't cross the membrane until it uses malate, aspartate, or glycerol 3-phosphate shuttle.
Yeah. Let's see. Okay, I think the best way to think about it is, let's see. I think the reason why it doesn't work is because NADH and like NAD+, they're very big and bulky. And also they're, in the case of NAD+, it's like also charged. That makes it hard to pass through the membrane. So I think essentially the only thing that's really of note is just, you know, the NADH or the NAD+, like that's gonna have to be converted into other molecules or added on to other molecules in order to bypass that situation. So here we have NADH, and then we have malate dehydrogenase. So that's going to convert to malate, and then malate is able to cross the transporter between the intermembrane space and the matrix. And then once we're in the matrix,
NAD+ comes in again.
yeah, and it can basically reconvert back. Like basically, it's like you kind of have to break NADH apart
I think
and
it
then
makes sense.
reform
I'm
back.
just confused on why both are happening on both sides of the membrane. Like, why are both sides happening? Why are the same things happening on both sides?
What do you mean by like? You mean like this reaction and this reaction?
Like oxaloacetate, acetate, oxaloacetate.
Hi guys, don't you know?
Like the same shit, right?
We're done.
I think because
Is it just that these intermediates are able to pass through, and then that's the NAD+ for the
yeah, so the malate and acetate are the second. I think what's happening here is the malate that's going to convert back to oxaloacetate, and then in another reaction, we can aspartate can be a precursor to oxaloacetate. This is going to be something that you'll learn in like amino acid synthesis and everything. You don't have to worry about that for now. But I think the main point here is just that you have to basically convert these things into a form that can actually pass through. And so this is like the simplest way, like the human body decides that.
That makes sense. I guess I'm confused on what the context of this is, because this was from the oxidative phosphorylation thing, right? But I thought this was a lot of TCA stuff.
Okay, I think that's just a misordering of the slides then. But this is
What's the question?
I remember seeing this, and I feel like this was a lecture.
Yeah, this is TCA. So this one, or ET. Okay, so let's clarify.
Because we just did ATP transport, and then it went to this thing.
Yes, yeah. So NADH transport. NADH, remember in glycolysis, you form the cytosolic NADH. Yeah. NADH cannot go into the mitochondrial matrix on its own. It needs a shuttle transporter, or a shuttle pathway. And that's exactly what that is, where the oxaloacetate will combine with the NADH, reduce all that, and the malate now has that reduced energy inside
the
of it.
electrons?
Yeah, exactly. And then it goes inside into the matrix, and then it gets rid of the NADH through the Kreb cycle stuff. Does that make sense?
But does this have anything to do with like this shit
Yeah, of course, yeah. So remember ATP synthesis? If we use NADH, it forms 2.5 ATP, right? So it's efficient. Whereas if we use the glycerol 3-phosphate shuttle, it's faster but inefficient to generate ATP. So we form the FADH2, and that bypasses complex 1. So the end result is that you only form 1.5 ATP. So the relevance of this is if we need the energy right away, we wouldn't use this pathway. We would use this one because we need the ATP so bad.
But it's shorter and it's not as
efficient. Yeah. So you're losing energy, if that makes sense.
Okay.
Does that make sense?
Yeah, no, I remember. So this is a part of complex 2 or in between 2 and 3?
So it's very similar to complex 2. It's not the same exact thing because complex 2 is succinate dehydrogenase, right?
Yeah.
This one is a different one, but it acts similar, as in it uses FADH2, and it bypasses complex 1. So you can think of it as like complex 1, complex 2, and then that alternative one at the same place, and then complex 3, 4.
That makes sense.
Yeah.
Thank you, I appreciate it.
Yeah.
I'm gonna head out now, but yeah,
Yeah,
thank you.
yeah, you're welcome. All right. You guys ready for photosynthesis and gluconeogenesis? Oh man. Okay, so as you guys know, like I'm using the same slides, but I took out the ones that I thought weren't important. So if you ever get confused, obviously ask questions, but if anything needs to be explained more in depth, I spent a lot of time on the video. As you can tell, it's like 2 hours. So just refer to that if you ever need clarification. So we can start on photosynthesis right now. And I want to divide it into 2 parts. Obviously, we know there's the light-dependent reactions and the light-independent reactions. And what we're going to focus on is how do we even power our sugar generation? It's not like you can just grab molecules from within the plant cell. You have to get it from some external source. What is that external source? It's light from the sun. And so in order to convert it from light energy to chemical, we have to do these light-dependent reactions, which involve pigment molecules. They involve like a bunch of like conjugated pi systems. I'm just giving you a brief overview of what we're going to be talking about. And so essentially what we're going to do is convert the carbon dioxide and the water, these very basic elements, use them to form our oxygen, and then on top of that, our sugar molecule. And yeah, so going into it, we're going to talk about the light-dependent reactions. We know that the chloroplasts contain, it's the site of photosynthesis. We have there's 3 layers. So we have the outer membrane, the inner membrane, and then the thylakoid membrane. And we know that the Calvin cycle occurs in the stroma, whereas the light-dependent reactions occur inside the thylakoid lumen, thylakoid membrane. So that's like a distinction between compartments that you should know. It's a little bit different and confusing than the mitochondria, but we will break that down. And something I want to emphasize about the thylakoid membranes is that the protons themselves, remember that we're still using that proton motive force, like in ETC. But in this case, instead of the protons being pumped across the membrane and then having to be pumped back, we basically bypass that, and ATP synthase reverses directions. So now the actual rotor is facing inside. So it's like more conserved, if that makes sense. And I will have slides on it. I'm just telling you ahead of time. It's slightly different in terms of the orientation of the enzymes compared to ETC. So let's talk about, and that's why I drew this diagram here. I really dislike this diagram because it's insinuating something that I just don't like. But so the first step that we're going to have, reaction 1, is light is going to come in, and we have these antenna molecules. These are our pigment molecules that are going to be able to take the light energy, these photons, and convert them into electronic energy, also known as excitons. And so that's exactly what we see in this first step, where we have the light shine into the antenna molecule. It captures that energy and excites the electron that it already has. Or it's going to basically it's an exciton that gets excited. It comes from the light. So the electron is excited, and it's going to transfer onto nearby antenna molecules. And if we were looking at this diagram up here, all it is it's just going from the outside to the reaction center. So it's making its way towards the reaction center. And then as it makes its way and it gets near the reaction center, what's going to end up happening is the energy is going to transfer onto the special pair. Now, and I want to make this simple and not use abstract terms, but the photocenter, what I'm going to be referring to is P680 photosystem 2 as our reaction center, our special pair. So this blue, like light blue right here, is going to be P680, is how I'm going to represent this. So the electron is going to be transferred onto photosystem 2, and photosystem 2 ends up getting excited. Now, this is the reason why I very much dislike this diagram, is because this part right here from step 4, where we go from the electron being excited in the blue to the red, the electron acceptor, it's almost insinuating that it's going even higher in energy, which that just doesn't make sense because we're not adding any more light to it. What I was trying to draw with this diagram is we have our electron acceptor, and it's going to get excited, right? But it's going to stay degenerate. It's going to stay at the same level. It's not that it goes any higher. And if anything, it goes slightly lower so the electron doesn't go back. And this is what this diagram is just showing right here. This is our reaction center, this P680 star, and it's going to transfer the electron straight to the electron acceptor, which is our pheophyton. And it's actually just technically a little bit lower, and that's because we don't want the energy to go back, and it keeps going down the reduction potentials. It's very similar to like ETC. If you guys remember how it goes from NADH all the way to oxygen, but it never goes back. We never see like a backwards direction. It always goes down because the energy decreases ever so slightly each step of the way. It's a similar thing here. So that's why I really like dislike this diagram, but hopefully that like makes sense.
Would you be able to point on this diagram like what part lines up with the carbon?
Oh yeah, it's sorry, I was kind of jumping the gun a little bit, but it's on this top left part where we have photosystem 2 being this light blue. We have our pheophyton is the red right here, and then we have our H2O, which I will talk about right now. So after does that answer your question, by the way? Okay. So basically, once we donate that electron, right, from the reaction center, from photosystem 2, it gets transferred onto pheophyton. We're going to have this empty P680, but we need to add electrons there in order for it to get excited. So how do we do that? We use an electron donor. And it's not necessarily that it's lower in energy, it just hands its electrons over. And so to orientate you within this diagram, we're going to be extracting the electrons from water and passing them over to photosystem 2, which is our light blue area, our reaction center, our special pair. Does everyone get that, at least right now? So if we're doing this reaction where we're transferring that electron, we end up basically replenishing the missing electron. And that's how this whole like diagram works. Hopefully that makes sense. And just now talking about more so this diagram, forgetting about the older one. So basically we have light come in, and we're going to start at photosystem 2. Photosystem 2 is excited at a wavelength of P680. And just going a little bit further ahead, P680 is a lower wavelength than P700. The reason why that is because a lower wavelength correlates to higher frequency. It takes a greater energy to excite this photosystem. So obviously, when it's in the middle of the day, this is just like an example, follow me, where everyone tells you wear sunscreen because there's UV rays in the middle of the day. We know that UV is lower wavelength, higher frequency. And so that's why in a few slides, I mentioned that photosystem 2 is activated much faster than photosystem 1 because we have those UV rays, because our lower wavelength is more prevalent and it's able to excite photosystem 2. So I was just going a little bit ahead, but we'll go back to this diagram. So with photosystem 2, P680 gets excited when the wavelength is 680, and then it passes its electrons onto pheophyton. And then from pheophyton, it goes straight to PQA, PQB. It's plastoquinone. You don't really need to know the sub-isoforms, but you can just memorize that there's two. I don't know. You don't really need to know that. And then what ends up happening is we pass those electrons to cytochrome B6F complex. And cytochrome B6F complex, you can think of it very similar to complex 1 of the ETC. So there's a lot of parallels, but this complex right here, B6F, will pump protons and start forming that proton-electron motive force. And that's what contributes to our ATP gradient. So every time we pass to the cytochrome B6F, we contribute to the proton motive force.
Can you clarify where the special pair is in the photosystem?
It's the P680. That's our special pair. It's this light blue. Whereas so basically, let me clarify. The reaction center, the reason why they use this diagram is because they're telling you that the reaction center is where everything is spatially coupled. It's very close together, so it allows for that fast transfer. That's what this diagram is trying to show you. So when you see this battery of like the 3, it's not that they're separate energy levels. It's the fact that they're so close together that it'll transfer the electrons really fast. Does that make sense? So when I and I'm like misspeaking a little bit earlier, but when I say reaction center, I'm talking about all 3 of them being spatially coupled. But when I say special pair, that relates to photosystem 2. And photosystem 2 is the thing that gets excited and all that. Does that make sense? So cytochrome B6F contributes to the proton gradient. What ends up happening is the electrons get passed on to plastocyanin. And from there, if we're now extrapolating this diagram on the top left to photosystem 1, our special pair, the light blue, is going to be photosystem 1. It's going to be P700. And the red, our electron acceptor, for this like bar over here for photosystem 1 is going to be our A0 like ferredoxin. You can just say it's ferredoxin. And then for the dark blue, instead of it being the water, it's going to be our plastocyanin. That's our electron donor. So you can see how it's adapted essentially to photosystem 1. So P700 is going to grab the electrons from plastocyanin, but the electrons on their own are not just going to jump up in level. We have to introduce light again to excite it. So that's actually what happens here is the plastocyanin says, hey, take these electrons, and P700 says, I'll accept them. And the light comes in and says, and basically increases the energy of it. And so that's what we see this drive up in energy, if that makes sense. Does anyone have questions about like that little process where the electron comes in to donate to P700, and then the light comes in to excite that and basically raise it in energy. Yeah.
Could you say the electron acceptor, electron donor, and special pair for flow system 1 again?
Yeah, of course. So the special pair, instead of P680, is going to be our P700 right here. And then our electron acceptor, just think of it as ferredoxin. It's going to go through cofactors. Just think of it as ferredoxin. And then our electron donor is going to be our plastocyanin. Now, it's not going to be water. Okay, and then and we'll go into like each of the photosystems. But photosystem 1 will transfer the electrons through A0, which I don't even know what that is, so it means that you don't really need to know. But it's going to use iron sulfur clusters and then pass it on to ferredoxin. And now ferredoxin can go 2 separate ways, right? We know that there's the cyclic and the non-cyclic. So ferredoxin can go through the cyclic, and it'll basically make a loop all the way back and go to cytochrome B6F, and again, contribute to the proton gradient. Yeah.
Okay, so is the I guess the jumping's like the same happening 2 and 1, but like basically you just change out like the donor versus acceptor.
Yeah.
And then is it fair to say that Z scheme is like the version of electron transport chain in plants, or like is that?
The Z scheme just talks about how the energy is excited and transferred. So
Z scheme just referring to the graph itself.
Yes, exactly. So it gets excited, then it goes back down as it gets transferred. That's why there's no back propagation. And then it gets excited again to form that last Z shape, and then it goes down. Yeah. So ferredoxin can go 2 ways, cyclic or non-cyclic. We talk about cyclic. And then the non-cyclic, we see here that it's ferredoxin NADP plus oxidoreductase. So this is going to obviously be an oxidation reduction reaction. And they actually, to memorize this and make it simple, they give you the substrates too. So our first substrate is going to be our reduced ferredoxin, and it's going to be NADP plus, which is oxidized. And so they're going to do that electron transfer, and you're going to end up forming your NADPH. Is this all clear for photosynthesis? Okay, so for photosystem 2, going more into detail, I wanted to talk about the manganese, which is our cofactor. It's our metal. And all I wanted to orientate you is that it goes from a +4 state, meaning that there's no electrons, it has nothing, to a state of 0. So there's a reason why it can go from these 5 different states, +4, +3, +2, +1, and then 0. So when it's at the +4, we know that it can hold up to 4 electrons. It's 0 electrons, and that's exactly why we use 2 waters to occupy it and take it from +4 all the way to 0. So as you can see here, it's going to go the opposite direction, and now the electrons are going to get passed on to the special pair, and then it's going to do its thing in photosynthesis. So that's what the manganese does. It helps coordinate the electron transfer from the waters and the electron donor to the special pair, that P680 in this case. Does that make sense? Okay. And so that's why we use 2 water molecules, because there's 4 electrons if we were, or in one water molecule, there's 2 electrons, multiply that by 2, there's 4. And that's mainly what I wanted to say. And also another thing, plastoquinone is very similar, not the same, but similar to quinone, or I forgot the name, ubiquinone, and in that it stays in the plasma membrane. So when it goes from photosystem 2 to cytochrome B6F, it's all in that thylakoid membrane. And then this photosystem, something to note, because they may test you on it, they may ask you, why is it named photosystem 2 or photosystem 680? Like, how does that relate to the energy of the light? And as I mentioned, like basic physics, is that shorter wavelength means higher frequency, and we know from the formula E equals HF, frequency, if you increase the frequency, the energy also goes up. So something to keep in mind. With photosystem 2, we need more energy to excite it. Any questions about what I just said? It's fine if I can repeat it. Okay. So moving on to cytochrome B6F complex, we're basically transferring the electrons from plastoquinone. So if I go back to this diagram, our plastoquinone all the way to plastocyanin. So that's where we are orientating ourselves. And it undergoes the Q cycle and basically leads to proton gradient, but the protons are pumped into the thylakoid. And so I say it's efficient because now it just goes back the opposite direction, but we'll explain that in a little bit when I talk about ATP synthase in this case. And the cytochrome B6F contributes to the gradient to power ATP synthase, and ATP is needed for our carbon assimilation reactions, the Calvin cycle. The Q cycle is very similar to the other one, but I won't talk about protons or any of that until the very end, the very end of this slide sequence. I have a slide dedicated to that. And then in terms of photosystem 1, we now just transfer the electrons to plastocyanin. Now what we're going to do is, again, go through this reaction center again, but instead we're going to change the names of the molecules accordingly. So photosystem 1 is going to grab its electrons from plastocyanin. Now, a key thing that I didn't mention is that plastocyanin is very similar to the cytochrome C in ETC, meaning that it only carries one electron. It doesn't carry two. So then the plastocyanin donates its electron to P700. P700 gets excited by light. It's passed through these cofactors A0 and A1, I think. But the main important one that you have to know is that it's passed through iron-sulfur clusters. And then from the iron-sulfur clusters, it gets passed on to ferredoxin. And then ferredoxin is going to act as our key mediator. Does that make sense? The cofactors? Just really, realistically, the only ones you need to know are iron-sulfur clusters and ferredoxin. Or at least the electron flow, at least. And then ferredoxin, as we know, can go two paths. It can go the cyclic or the non-cyclic, which I talked about already. And then I wanted to talk about more the spatial biology of the photosystems. So photosystem 2 and photosystem 1 are in different areas of the thylakoid membrane. Remember that the thylakoids, they are pancakes, right? They stack on top of each other. So you're going to have areas where the two adjacent thylakoid membranes are basically on top of each other, and they're very close. Now, this is key because photosystem 2 is found in those regions where the pancakes are over each other, where there's overlap, or basically it's called oppression, I think. But photosystem 2 is in the oppressed portions. So as you can see in this diagram, it's these like purple, light blue, whatnot. It's these. And the reason why and I'll explain why this is important, but for photosystem 1, they're not found within the pancakes. They can be found on the sides where they're not oppressed, or they can be found on the very, very top where there's nothing, or on the very, very bottom where there's nothing. And there's a reason why. Remember that photosystem 1 is going to transfer its electrons to ferredoxin, and ferredoxin can go the non-cyclic pathway and transfer the electrons to NADPH, right? Well, NADPH, if you guys look up the structure, it is very large. So that's the reason why physiologically or spatially why we have photosystem 1 in the non-oppressed regions, is that the NADPH is so big that it needs to get close, and it wouldn't be able to do so if it was in these oppressed portions right here, right? So that's why you see photosystem 1 in these regions right here. And I also wanted to orientate you to the light harvesting complex here, where it basically acts as our antenna molecule. If you remember from this diagram, these green molecules, you can think of them as our light harvesting complexes. And what's a special property about these light harvesting complexes is that they are able to be phosphorylated. And this is something to note, is that they are phosphorylatable through their threonine residues. And we know threonine has these OHs, and obviously they can be phosphorylated. You can attach a phosphate group onto it. And what that allows it to do is basically like have an electrostatic repulsion from the plasma membrane and cause this light harvesting complex to move from photosystem 2 to photosystem 1. And remember, the light harvesting complexes want to pass that electron energy to the nearest thing that it can. And so if it moves closer to photosystem 1, like this diagram right here, it's going to favor that electron transfer to photosystem 1. Your question? Yeah.
Why are the threonine residues in the intramembrane space?
I think that's just like a cartoon diagram, like honestly. Yeah. Any more questions? Okay, so when the threonine residues are phosphorylated, you can just think of it as the phosphate group is such a larger group that it's going to basically open up the thylakoid membranes and cause that light harvesting complex to move. Whereas if it's the hydroxyls, they're not as big, so of course the thylakoid membranes are going to come closer. And so that's just something to note, is that during the day, the light harvesting complexes are phosphorylated and they're moved towards photosystem 1 because there's that buildup of these plastocyanins, PQH2, plastoquinones. And yeah, you guys should definitely memorize this cascade. Basically what I just said, accumulation of PQH2 leads to activation of these protein kinases. They phosphorylate the light harvesting complexes, which allow for the light harvesting complexes to migrate towards the photosystem 1 and increase the activation. So it's almost like this fine balance of activity. And then once there's a balance, or when it turns nighttime, we basically activate the phosphatases and they dephosphorylate it. Okay, and then just like a brief overview about like protons and ATPs and everything that you should know is when you're doing photophosphorylation, for a single molecule of O2, we're going to form 3 ATPs and 12 protons. Now, a lot of times we like to show the diagram of one water, which forms half that oxygen molecule, but like I don't really like to think of it like that. I like to think in whole numbers. So for half of that oxygen molecule, if we're just talking about the one water that transfers its electrons, it would be 1.5 ATPs produced and 6 protons across the membrane. And that's exactly what this is showing you right here, this diagram. As you can see, we're talking about the one water and then the half O2. And so the 2 protons from the water are going to get transferred, are basically going to get pulled apart from the oxygen. We're going to form our oxygen gas, and then the protons are going to go into the lumen side of the thylakoid and contribute to the ATP synthase. And then our last 4 are going to come from the Q cycle occurring twice, essentially, because we have it occur the first time, 2 protons are pumped, but we remember that plastocyanin only carries 1 electron, so we have to do the Q cycle one last time. It's a two-part reaction, and then the last 2 protons are pumped. So that's our 4, that's our 2, and that combines to form 6 protons across. So honestly, you can just literally memorize the bold and you'll be fine for the exam, but just wanted to describe it. So how do we feel about like photosynthesis? Any questions, anything that pops up? Because we're going to talk about Calvin cycle next. Okay. So talking about the Calvin cycle, we're now talking about the stroma, right? And we're not in the thylakoid lumen, we're not in the thylakoid membranes, we're just in the stroma. You can think of it, well, actually I don't want to even do comparisons. It occurs in the stroma. So basically the purpose is to convert CO2, or carbon dioxide, into sugar molecules. And so we're going to go through 3 parts. We're going to go through fixation, which is we force that CO2 in, and it's not a carboxylation, and I'll explain why in the next step, but it's not a carboxylation. So fixation is a special type of stage that the plants can do that like humans obviously cannot. Then stage 2 is the reduction, where we turn our 3-phosphoglycerate into a glyceraldehyde 3-phosphate. And then from there, after 3 carbon dioxides, 1 glyceraldehyde 3-phosphate can leave. And then the rest of them get converted into ribulose 1,5-bisphosphate through stage 3, which is regeneration. So the key intermediate is ribulose 1,5-bisphosphate. It's going to allow the cycle to keep occurring. And essentially, like the motivation of this whole pathway in the first place is that we know ATP isn't a good energy storage molecule. And also, like if the plant needs it in the rest of its parts, ATP can't necessarily cross the plasma membrane or whatever to go into the blood flow and then all that. We know that doesn't happen. ATP isn't just in your blood, it's in the cells. And then this is something also I want to, if you don't understand necessarily where the ATPs and ADPHs come from, just memorize what's in the red. For every 3 carbon dioxides that are fixed, we form our 1 glyceraldehyde 3-phosphate. We form 9 ATPs to produce that, and then we use 9 ATPs and we use 6 NADPHs to get that 1 glyceraldehyde 3-phosphate to leave. So if you ever get confused on the countings, just memorize this and you should be good. Yeah. Okay. So now moving into the stages. So I really want to emphasize that this is not a carboxylation reaction, because carboxylation reactions, we know that they typically occur on ends of a molecule, right? So we'll typically have a carboxylation group added to the end, whereas in this diagram right here, what ends up happening is we have our carbon dioxide that's being added into the middle. So it's not necessarily carboxylation because you're adding it into the middle. You're doing some special reaction. You don't have to know the mechanism. That's why we call this carbon fixation rather than carboxylation. And the way we do so is through this enzyme called rubisco. It's a very slow enzyme. So because and I don't want to go like back to the Michaelis-Menten, just know that it's very slow to convert the ribulose 1,5-bisphosphate into the 6-carbon molecule. It's very slow during that reaction. So we just need to add a lot of the rubisco enzymes, and then now it's productive, supposedly. So that's like the compensatory mechanism, if that makes sense. And again, it's similar activity to biotin, except biotin will carboxylate, whereas but it's very similar to biotin, but it's not the same because biotin only binds to CO2, but rubisco can bind to CO2 and O2. And you guys know about O2, which is like the C3, C4, CAM, photorespiration. We'll go into that, but that's one thing to know, why it uses O2. And so at the end of this first stage, we end up forming, for for one carbon dioxide incorporated, we end up forming 2 3-PGs, if that makes sense. So our 3-phosphoglycerate, there's 2 of them formed for one single carbon dioxide. So that's something to know. That's why at the end of it, if we do 3 carbon dioxides, we end up forming 6 of these 3 PGAs. And then for stage 2, there's reduction, where we convert basically our 3 PGA into glyceraldehyde 3-phosphate. First step we do is phosphorylate it, and then after that, we end up reducing it. And so in this step, again, remember in this previous one, the fixation, if we're using 3 carbon dioxides, we're going to end up forming 6 of the 3 PGAs, right? So this is technically multiplied by 6, and we end up with 6 glyceraldehyde 3-phosphates. And that's where, again, for each single one, we use 1 ATP and 1 NADPH, but we just multiply that by 6, and then we end up forming our 6 G3Ps. And I think that's it. And then basically only 1 G3P is allowed to leave. The other 5 have to be used in the regeneration step. So I'll talk about the regeneration. No, you don't have to memorize the enzymes. Don't do that, please. That's just going to be a waste of your time. What I will emphasize is know the red rectangles and the purple rectangles on this slide. And what I mean is that just count the number of red rectangles there are. There's 5 3-carbon molecules, right, that go into this regeneration phase. And then what we get out are 3 5-carbon molecules. So in this case, I know there's 2 pink rectangles. It's like this diagram I really hate, but it's essentially step 9 occurs twice. So you can count this purple rectangle or magenta, whatever, as times 2. So 2 ATPs are used here. The third one is used here. So for the regeneration phase, if we are incorporating our 3 carbon dioxides, we end up using 3 ATPs. Hopefully that makes sense. Does anyone have questions about like Calvin cycle in general, at least in terms of like the net ATPs, net NADPHs? Okay. And then the reason why I bring this slide up is because, so what if we get rid of our one glyceraldehyde 3-phosphate that leaves? How does it get out of the chloroplast? Because it's inside the stroma, right? That's what the shuttle is for. And essentially, all right, did I skip it? No, okay. So essentially what we have here is our glyceraldehyde 3-phosphate on the stroma side, right, on this diagram. So the glyceraldehyde 3-phosphate is going to go from the stroma to the cytosol and then participate in glycolysis, gluconeogenesis. It needs to be transported first, and that's what this thing is, this shuttle. So our glyceraldehyde 3-phosphate will get converted to dihydroxyacetone phosphate, and then from there it's going to go through this, it's inorganic phosphate triose phosphate antiporter. You can just, you can call it that, or you can call it DHAP, inorganic phosphate antiporter. You can do either or. But essentially, dihydroxyacetone phosphate goes from the stroma to the cytosol, and then the inorganic phosphate goes in the opposite direction inside. And then from there, we end up using, again, this is a glycolysis enzyme, the triose phosphate isomerase, and we convert it from DHAP into G3P. And then from G3P, we can either go to glycolysis, we can let it go through this transport, we can let it go through gluconeogenesis. There's a lot of things we can do now with this, and it can diverge in multiple pathways. So this is a transporter that can feed into multiple of these different pathways. And then one thing I want to, or actually this, and then these, what I didn't mention before, is that in the stroma, in these steps right here, these 3 molecules, this is our step 2, stage 2 of Krebs cycle. So this is our fixation, or sorry, not fixation, reduction, where we add the phosphates and then we add the NADPHs. So if I go back to this slide right here, we see the reduction of ATP, or we add the phosphate and then we end up reducing it. That's exactly what's happening here with the 3-phosphoglycerate, the 1,3-bisphosphoglycerate, and then the glyceraldehyde 3-phosphate. That's exactly what's happening here. Does anyone have any questions about this, like transporter or anything like that, or how it fits into the whole thing? Okay. And then one important point is that the Calvin cycle is indirectly activated by light. So we know that if light is hitting the, so if we know that the light is hitting the thylakoids and they're basically activating photosynthesis, our light-dependent reactions, we know that protons are going to get pumped into the lumen. So if protons are pumped into the lumen, that means that our pH is going to decrease because the proton concentration goes up. So inside the lumen, pH decreases, and in the stroma, because the protons are leaving, if we were to take it from before light was activated to now light being activated, in the stroma, the proton gradient decreases because those protons are going into the lumen. So the pH of the stroma ends up going up because we decrease the concentration of the protons. Those protons are going into the lumen. And then also another thing to note, and this is just memorization, I don't have an explanation, is that magnesium will increase in the stroma. The concentration of it increases when light is present, when light is activating the light-dependent reactions. I don't know why. I just know that's something that I had to memorize, and so I put it in the slides. And then just like a fun fact, in light activity, Calvin cycle activity increases more than 100-fold. So you don't have to memorize the 100-fold. You just have to know that Calvin cycle increases activity in light. And it just makes sense because if we try to make the Calvin cycle go in the dark, where are we going to get the energy from? Where are we going to get the ATP and the NADPH if it's dark? We don't have the light to power it. So that's why it occurs faster in the light, because we have that energy coming in. And we can go through the Calvin cycle much more frequently without having to worry about having unused intermediates. So remember how I mentioned that Rubisco is a little bit different than biotin. Biotin can only incorporate CO2, and Rubisco can incorporate CO2 and oxygen. So this slide and the next few are going to talk about what happens when we incorporate oxygen. And first thing I want to preface is that no, you do not have to memorize all these intermediates on this diagram right here. This is not worth your time at all. The only thing you really need to know is that it ends up using 1 ATP to convert our wrong product, the 2-phosphoglycolate. That's not what we want. We wanted the 3 PGA, whatever it was. When we create this thing right here, we have to convert it to our 3 PGA. And so we go through this pathway through multiple organelles. You don't need to know all the enzymes. And just know that we end up using 1 ATP to convert it to 3-phosphoglycerate. And that is costly because we're using 1 ATP. So instead of using, I think it was like 9, we're using 10. But don't obviously I can check after that. But in terms of this, it uses 1 extra ATP. And it's just a long salvage pathway. And this is for C3 plants. So in the next slide, I will talk about the C4 plants. And so I just want to orientate you to this diagram right here, where we have 2 separate compartments now. This is an evolutionary adaptation in C4 plants. We end up having a mesophyll cell and a bundle sheath cell. You should memorize the names of those cells and what each compartment does. So in the mesophyll cell, we know that carbon dioxide and oxygen can get in. Now what ends up happening is carbon dioxide goes through this reaction or whatever and gets incorporated into 4-carbon inorganic or organic acid. So basically, carbon dioxide gets fixated by a different enzyme, and then because it forms this new fixated organic acid with the carbon dioxide, it's going to be able to transfer from the mesophyll cell and basically translocate into the bundle sheath cell. So it's able to move through. Oxygen cannot do that, and it cannot get incorporated. So that's a way to separate the 2 compartments, and that way you don't have the wasteful oxygen going into your Rubisco enzyme. And so the Rubisco is going to be concentrated in our bundle sheath cells. So that's the whole function of C4. That's the adaptation. We spatially separate the compartments so oxygen doesn't get wrongly incorporated. Does that make sense? Okay. And then in terms of the CAM plants, instead of using spatial compartments, we're talking more about temporal aspects of it. So in the night, the plant cells will open their stomata and carbon dioxide will come in. So whenever a plant cell, just like general plant physiology, if it opens its stomata, stuff can come in and out, like water, carbon dioxide, oxygen, it does that. So if you activate it in the day, obviously oxygen is going to come in. But the CAM plants have basically genetically evolved to only open their stomata at night. And at night, we won't have the light reactions going, right? So oxygen won't be created through the light and the light-dependent reactions, right? Because we know that water will turn into oxygen. So at the dark, it'll open up the stomata and carbon dioxide will come in. And then what ends up happening is during the day, we have such an abundant store of carbon dioxide that it'll get incorporated through the Calvin cycle. And this is just an evolutionary adaptation to allow for less of the photorespiration to occur. Does that make sense? Okay. And that was all of photosynthesis and Calvin cycle. Do we have any questions about just generally anything that popped up, anything like that? Okay. So talking about gluconeogenesis and glycogen metabolism. So I'm going to break down the regulation stuff because I know that is very, very, very confusing. And I had a hard time understanding this actually when I was reviewing it. So hopefully I can explain it very well. So basically gluconeogenesis, as you all know, it's in the name, neogenesis. We're forming new glucose. And we're basically making it from pyruvate, from some glycolysis intermediates. It's mainly from pyruvate and some other like Krebs cycle intermediates as well. But realistically, you only need to know that, or start from pyruvate is like the general scheme of things. And the goal of this is that we want to convert our 3 carbon molecules, 4 carbon molecules, and we want to turn them into 6, into glucose. So that's the whole point of this. And I wanted to emphasize, triacylglycerides, tags, cannot be used to generate glucose in vertebrates. And that's because tags will essentially undergo beta oxidation. And I don't want to confuse y'all. All you have to know is that tags will basically break down into acetyl-CoA. So we have like we have like a fatty acid chain, and all it's going to do is we're going to just cleave every 2 carbons. So this will form our acetyl-CoA, this will form our acetyl-CoA, and that's all you need to know. So that's why tags don't form they're not gluconeogenic, because, again, remember, acetyl-CoA will go into TCA and then come out as carbon dioxides. They will never replenish the TCA intermediates. So that's exactly why tags can't actually generate glucose. No, tags are like fatty acids. Like you can just think of that. They're long chains. And when you guys haven't learned this yet, I'm just giving you like a reason why. So honestly, you can just memorize that tags can't be used. But tags, you can cleave them every 2 carbons, and they end up forming your acetyl-CoAs. And you'll learn that in like the next 2 lectures or whatever. But I just want to tell you that it breaks up. Oh, it's Thursdays? Well, I mean, it's not an exam. So like, it just breaks down to acetyl-CoAs. That's why. And gluconeogenesis occurs mainly in the liver. Remember that the liver has hepatocytes, and they interact with blood glucose. They act as like blood glucose sensors, that organ. And it'll be the site where we generate glucose in case that we have low blood glucose levels. So this mainly occurs in the liver, not necessarily like tissue cells. It can, but it's like skeletal muscle can also have gluconeogenesis as well, but that's skeletal muscle. Okay, so gluconeogenesis is not just a reversal of glycolysis. We know that there are 3 steps in glycolysis, steps 1, 3, and 10, that are irreversible. And so it's steps 1, steps 3, and step 10. And so we have to undergo a bypass reaction. And so we'll talk about the more complex one first, which is bypassing the last step number 10 in glycolysis, where we go from pyruvate to phosphoenolpyruvate. Yeah. So for the first bypass, there are 2 parts to this, and I'm just showing you right now, like very quickly, that they're slightly different, and the only reason why is because they occur in different tissues. For this first one right here, it occurs in tissues lacking lactate, so meaning the liver. We know that muscle cells produce lactate, skeletal muscle. So in this other one, this is where we have the lactate present. So we'll go into each one in depth, but I just wanted to emphasize that there are kind of 2 different pathways for the bypass reaction, depending on which tissue you are. So for the first one, we have to transport pyruvate into the mitochondria, and then from the mitochondria, the pyruvate is going to react with this new enzyme. Now that's specific to gluconeogenesis. It is pyruvate carboxylase. Now I mentioned before that biotin is a very special cofactor, coenzyme, whatever you want to call it, and it incorporates CO2s into the molecules, and that's exactly what we see here, where we have our CO2. We use up an ATP, and we end up forming our oxaloacetate. Remember, oxaloacetate is a 4-carbon molecule. Pyruvate's 3, so it makes sense that if we add 1, we end up generating our oxaloacetate. When we generate this, we end up forming our NAD. We end up grabbing the NADH and reducing oxaloacetate into malate. And then this is very reminiscent of the malate aspartate shuttle right here. These 4, the oxaloacetate, the malate to oxaloacetate. This is very similar to that malate aspartate shuttle. This is just not necessarily like a circle that we've seen before in those classical diagrams. It's just a linear. And so when it converts to malate, it goes to the malate, a malate alpha-ketoglutarate antiporter. And then it'll basically go into the cytosol. The malate will then release the NADH cofactor, and then it'll form into oxaloacetate. And then we have this cytosolic PEP carboxykinase, enzyme number 2, that's crucial for this bypass reaction, right? So oxaloacetate will basically be decarboxylated, and in lieu of getting rid of the CO2, we're adding a GTP, a phosphate group to it. And there's a reason why we have these 2 iterations, now that I mentioned that, is that for the tissue lacking liver, our cytosolic PEP carboxykinase, for tissues lacking liver, our PEP carboxykinase is only found in the cytosol of the cell, whereas in skeletal tissue that generate lactate, our PEP carboxykinase is only found in the mitochondria. So that's the distinction why we have these two different pathways in the first place, is that the PEP carboxykinase is in different regions depending on the tissue. So that's something you just have to memorize. I don't know necessarily the physiology of it. But yeah, and then basically at the end of this bypass reaction 1 in liver cells, we end up forming our PEP, which then goes through upwards in the reversal direction of glycolysis. Is everyone clear, at least on this first part? Okay. So then for the second half, right? Now our PEP carboxykinase is inside the mitochondria, right? It's right here. And what we have to do is, instead of using this malate aspartate shuttle, we have to utilize the pyruvate transporter, right? So the pyruvate can go into the mitochondria along with a proton. That's one thing that allows pyruvate to enter the mitochondria. And then from there, it gets carboxylated to oxaloacetate. And then from oxaloacetate, it'll be converted into PEP through this. And then PEP can, I think PEP, I don't know, you don't have to know the transporter. You don't have to know the transporter, but it's basically going to translocate from the mitochondria into the cytosol, because we know gluconeogenesis occurs in the cytosol. I can look that up for you all after like this review session as well. And yeah. And then in terms of why we talk about lactate generating tissue is because, we have to avoid shuttling NADH out of the mitochondria. So when we have lactate dehydrogenase, remember that it's also a NADH generating. And we talk about it in terms of anaerobic respiration, where it goes from pyruvate to lactate to form our NADH. So I'm just going to write P for pyruvate, it goes to lactate, and then we end up forming our NAD plus, which allows glycolysis to keep occurring. And that's exactly why we have lactate dehydrogenase in skeletal muscle, because skeletal muscle needs ATP right away. And if it has to rely on anaerobic respiration, it will. And whereas the liver, it's very heavily oxygenated. I don't know if you guys know about it's the portal vein and all that stuff. But there's like a whole new circulatory system that goes straight to the liver to supply oxygen to it. So that's what the subtle distinction between the two. Is everyone good on the first bypass reaction? Any questions about it? Okay. So in terms of the regulation of this first bypass reaction, remember that we talked extensively about PDH and how it's heavily regulated and inhibited when we have a buildup of energy, we have a buildup of acetyl-CoA. So what ends up happening is acetyl-CoA can actually act as an activator for pyruvate carboxylase. And so acetyl-CoA will stimulate our pyruvate carboxylase because we want gluconeogenesis to occur. We want that to happen. And so pyruvate carboxylase, we see that in both of these diagrams, they're inside the mitochondria. So it's not like the acetyl-CoA is leaving or anything. It's inside, so it can interact with it. And so it'll basically activate the pyruvate carboxylase. It'll promote this bypass reaction, and we see that gluconeogenesis is favored when we have high acetyl-CoA, meaning that the Krebs cycle is backed up. Does that make sense in terms of regulation? Okay. Oh, I think I skipped the next one. I didn't get that slide. Okay. Let me get that slide, actually. I don't know why I don't have bypass reaction 2. Okay, bypass reaction 2.
What would that mean for glucose levels and energy levels is the Krebs cycles?
I will talk about the difference between the two, because I know that's the most confusing part. But I will talk about in terms of glucose energy, if we have, like, for example, low glucose, but we have high energy low energy, what ends up happening. I'll talk about the distinctions. In terms of bypass reaction 2, so I didn't include this in the slides, but essentially we end up having our fructose 1,6-bisphosphatase. This is our new enzyme, right? And this is going to basically take our fructose 1,6-bisphosphate, and what it's going to end up doing is it's going to FBPase1 is going to dephosphorylate fructose 1,6-bisphosphate at the 1-phosphate carbon, if that makes sense. That phosphate group that's attached to the carbon number 1, it's going to dephosphorylate that, and it's going to form our fructose 6-phosphate. And what it needs as like a prerequisite is water to come in. It needs to hydrolyze that phosphate bond. And so that's why we use the one water, and then we end up forming our inorganic phosphate that leaves. And this step doesn't require or generate ATP at all, because it's just a hydrolysis reaction. And I think that's that. Yeah, okay, so I'll go back to the other slides now. Sorry about that, I should have included it. And then in terms of bypass reaction 3, this is the final step of gluconeogenesis, first step of glycolysis. So we know that we're going to use a glucose 6-phosphatase. It tells you exactly what the enzyme is doing. It's taking your glucose 6-phosphate and getting rid of the phosphate group. That's it. And then we end up forming our free glucose, which again is now able to transport across the plasma membrane using a glucose transporter and go into the blood and eventually increase our blood glucose levels. And again, this one doesn't use ATP. It's very similar. It uses water as a prerequisite to hydrolyze that phosphate bond. But yeah, those are the 3 bypass reactions. Does anyone have like questions for this? Okay. And then I just wanted to reiterate glucose. Oh, sorry. Go ahead. It's in this step right here. So we end up using 2 ATP here, right? And this is multiplied already, accounting for the 2 pyruvates that are going to combine together. So we end up using 2 ATPs on the very bottom right for the first step, right? Because we're phosphorylating it. And then in the pyruvate carboxylase phase, because biotin ends up using up ATP in order to carboxylate. That's why we use ATP for the first step. Then we use our 2 ATPs from converting 3 phosphoglycerate into 1,3-bisphosphoglycerate. That's where our last 2 ATPs come from, and that forms our 4. Does that make sense? Yeah? Okay. And so I wanted to reiterate, gluconeogenesis is very expensive. We end up using 4 ATPs, and we end up using 2 GTPs per molecule of glucose that we form. And remember, per glucose molecule, we have to combine 2 pyruvates, 2 3-carbon molecules together to form our 1 6-carbon. And so this gluconeogenesis pathway is very, very regulated using energy and glucose, and keep them separate on purpose. Don't try to combine them and make them all work together. Keep them separate because there's different conditions for such. And so ATP and AMP are key cellular regulators. They have, obviously, they're energetic. They tell you whether you have high energy or low energy. And then obviously correspondingly one or the other will favor the anabolic or the catabolic pathways. And then glucose sensing F26BP, fructose 2,6-bisphosphate, will also be used to regulate it, but in a different mechanism, not in an energy state, right? So if you have high amounts of glucose, think of it this way. F26BP will be at a higher concentration and only when you have high glucose levels. Actually, let me reword that. When you have high glucose, you're going to form your fructose 2,6-bisphosphate, and because you have high glucose, there's no reason for you to generate more glucose. So gluconeogenesis is going to get inhibited when you have high glucose levels. And we'll talk about this extensively now. So before I go into that though, I want to talk about the synthesis and breakdown of fructose 2,6-bisphosphate. So our fructose 2,6-bisphosphate uses isoforms of PFK1 and FBPase2. And instead of using PFK1, FBPase1, we just switch the number to 2. And that just correlates to where we're adding or taking away the phosphate group on the carbon backbone. And I think that's mainly what this slide says. And then the most important thing is that fructose 2,6BP acts as a glucose sensor. And so this is where we talk about the regulation. So we will first talk about the energy stuff. We will worry about the glucose in a little bit. So we know that energy, if we have high energy, we don't want glycolysis to occur. We see that in this diagram, ATP is going to inhibit, citrate is going to inhibit. Citrate you can think of as a high energy molecule if you want to, because TCA is backed up. So these two are going to inhibit our PFK1. And that means that glycolysis is no longer happening, and basically it's not affecting gluconeogenesis with the energy stuff. I mean, technically the acetyl-CoA, but the point is in this diagram, we see that high energy is going to inhibit glycolysis. Now, if we have low energy, ADP, AMP, they stimulate glycolysis because, hey, we need to generate energy. We need that to occur. So we're going to inhibit gluconeogenesis because it doesn't make sense for us to build up glucose molecules if we don't even have energy in the first place. That's why you see this dual reciprocal activation inhibition with AMP, where it activates PFK1 and it inhibits FBPase1. Now, I want to talk about the glucose sensor now. So this is separate. Don't even think of energy levels now. With the F26BP, it is only created when you have high glucose levels. If we have high glucose, there is no purpose for gluconeogenesis because we already have the high glucose. There's no need to form more. So what ends up happening is F26BP inhibits gluconeogenesis, and then it promotes glycolysis because we have high glucose, we want to use that energy. Now, let's say we have high ATP or high energy and also high glucose. What ends up happening? Well, the answer is that F26BP will stimulate glycolysis, but our high energy molecules inhibit. So what ends up happening is that glucose, instead of being broken down through glycolysis because there's high glucose concentrations, it's actually going to be converted to glycogen. So that's like the special condition that a lot of people get confused, is that when there's high energy and high glucose, we need to get rid of the glucose somehow. And how does it do it? It's through glycogen synthesis. And so this diagram doesn't show that, and you're stuck with like, oh, like glycolysis is also inhibited and activated. And in terms of if you have a high glucose, obviously you're not going to want to make more glucose. So that's not the issue at all. But does that make sense in terms of how to like think of these regulators? Keep them separate and then combine like the two conditions separately, if that makes sense. So whenever you're given a problem where it's like you have high energy and high glucose, what is the pathway that's favored? Just think, okay, high energy. I know glycolysis is inhibited, and I know gluconeogenesis, it doesn't really have an effect other than the acetyl-CoA. But we know that F26BP is a glucose sensor. So if we have high glucose, we know that we don't need to generate gluconeogenesis. We don't want to activate that pathway. But we know glycolysis is also inhibited by our high energy molecules. So then it goes to glycogen as a result. It has to go through some other pathway to get rid of that high glucose level. Hopefully that makes sense and I broke it down. But I know it was really confusing at first.
Can you try like just one more time?
Yeah, of course. So separating them, we know the energy stuff, right? If we have high energy, we don't want glycolysis. We don't want that. And if we have high energy, we can stimulate gluconeogenesis, right? Because we have the energy. But now that's that part. That makes sense. That's easy. Now let's talk about the glucose part, right? The F26BP. F26BP increases in concentration when there's a lot of glucose present. So when we have high glucose, our F26BP also increases. And what ends up happening is, just like rationalize it, gluconeogenesis makes glucose. If we already have a high glucose levels, we don't need to form it anymore. And then if we have high glucose, it would want to go through glycolysis. Now let's combine the two conditions. If we have high energy and high glucose, well, we need to get rid of the glucose somehow, because physiologically, to maintain homeostasis, we have to get rid of it. And so if you just think about the glucose itself, well, you would think, okay, it's going to go through glycolysis because we have high glucose levels. But that's not the case, because our high energy molecules inhibit glycolysis. And so the net result of the pathways is that you stimulate glycogen synthesis, because you have to get rid of that glucose somehow. And it's not going to be through glycolysis, it's going to be through glycogen synthesis or the pentose phosphate pathway or some other one, right? It's just not glycolysis is what I'm saying. Does that make sense? Okay. And then in terms of regulating our F26BP, it's a dual enzyme. So PFK2 and FBPase2 are connected together, like they genuinely are connected by amino acid sequence. What this means is you can only have one or the other on. It's like a light switch. You can either have it on or off. And so there are two domains on this peptide, and only one is active at a time. So PFK2, we're talking about this not in energy terms right now. We're talking about this in terms of glucose levels, because that's exactly how it's regulated. So remember, orientating ourselves, PFK2 is going to form our fructose 2,6 bisphosphate. So this is exactly how F26BP acts as a glucose sensor. That's what this slide is going to tell you. So in this one, PFK2 is going to be stimulated by insulin, right? And just to orientate you on this diagram, we see that PFK2 is active when it's hydroxylated, right? When it's not phosphorylated. Whereas FBPase2 is activated when we have the phosphate group attached, right? So that's just like a matter of fact right there. If we have a hydroxylated enzyme right here, this combined enzyme, whatever, if it's hydroxylated and OH group is attached, PFK2 is going to be the only activity happening. Now, if we have it phosphorylated, that dual enzyme, it's going to be the FBPase2. So now with that in mind, we're going to talk about insulin and how it regulates it. So just separating it from the slide, we know insulin is only secreted when we have high blood glucose levels. So we want to stimulate our F26BP, right? We want to create more because it's our glucose sensing molecule. So in this case, we want the condition that increases F26BP, right? And that means that from this diagram right here, PFK2 is going to be the one that we want active. So in this case, we want the hydroxylated version of this enzyme, right? We don't want the phosphorylated because this is the FBPase2. So if we have high glucose levels, if we have that insulin secreted from the beta cells of the pancreas, what's going to end up happening is our phosphoprotein phosphatase, right? It's going to be stimulated by insulin, and the phosphatase is going to dephosphorylate this enzyme, right? And when it's dephosphorylated, it's going to form this hydroxyl group. And when the hydroxyl group is here, we know that it's going to stimulate F26BP formation, right? That's the first condition. Does that make sense in terms of stimulation and activation by insulin? And then we'll talk about the other pathway now, the glucagon. So glucagon, just a little physiology background, is glucagon is secreted by the alpha cells of the pancreas, and it's only secreted in the conditions of, of low glucose levels, right? So when there's low glucose levels, glucagon is secreted because it's telling the body, hey, we need to create glucose, we're running low, we need the fuel. And the only reason why you're telling your body that is because your brain needs the glucose. That's its primary energy fuel source. So it communicates with the pancreas. But anyways, point is that glucagon will be secreted with low glucose levels. And what ends up happening is it stimulates our protein kinase. Now this protein kinase is going to phosphorylate this dual enzyme. So again, if we phosphorylate it, it's going to be the FBPase2 that is active. And when the FBPase2 is active, we're going to dephosphorylate our F26BP, this reaction right here. We're going to go from the double phosphorylated to the single phosphorylated when the FBPase2 is active, right? So in this instance, if we phosphorylate this enzyme, we end up stimulating the phosphatase activity. So that's exactly why glucagon or an increased CAMP is going to stimulate this enzyme and end up phosphorylating it. Does that make sense? Yeah. Yeah. It's very similar. I would just memorize it as the name. You could think of it as similar because, yeah, you're right. PKA is activated by CAMP. It is very similar. And I think that there could be a subtle distinction. Like there's like, I don't want to like confuse too much, but there could be like a CAMP dependent kinase that binds CAMP. And then the glucagon itself will bind to a metabotropic receptor, do that signaling cascade, and do PKA, maybe. But it's better to just think of it as CAMP kinase, if that makes sense. Yeah. But anyone else have any questions? I know this is like the more confusing thing. Okay. So shifting to glycogenesis. So remember how I talked about in the regulation stuff that if we have high energy and high glucose, the glucose has to go somewhere. Well, that's glycogenesis. So that's what we're going to talk about now. So when we're generating our sugar nucleotides, we have to use a different substrate. And the reason why we use a different substrate is because it gives us a different pool of reactants to work with. So remember this like recurring motif of like we have in glycolysis where one reaction is endergonic and then the other one is exergonic, and that pulls the reaction forward and it drives the reaction, the pathway to keep going. That's exactly the same principle with glycogenesis when it's stimulated. So we end up using UDP glucose as our substrate for glycogen synthesis. And in order to go from glucose to UDP glucose, there's a three-step process, right? We first convert the glucose to glucose 6-phosphate. That's very simple. That's the same enzyme as glycolysis. Then from glucose 6-phosphate, it's going to mutate, or basically it's going to like not necessarily isomerize, but it's going to transfer the functional group, that phosphate, from carbon number 6 to carbon number 1. That's the definition of a mutase, is it transfers a functional group. And again, the name of it is phosphoglucose. So you're dealing with the phosphorylated glucose. And then the third reaction, the final one, is that we're converting glucose 1-phosphate, or sorry, we're taking glucose 1-phosphate, adding a UTP, a uridine triphosphate. So it's a base. It's an RNA base specifically. If you remember uracil, that's exactly what this is. It's just a free uracil floating in space with a trinucleotide. That's what UTP is, for context. What we're going to do is take the glucose 1-phosphate, combine the UTP, and what's going to end up happening is one of the phosphates from the UTP is going to leave, and the UDP gets incorporated to glucose. And when that phosphate leaves, it's going to generate a pyrophosphate. Now, remember that we talked about in glycolysis the formation of ATP synthesis, where we end up when we end up forming our pyrophosphate. Wow, what am I saying? ATP hydrolysis. Sorry guys. When we have ATP hydrolysis, right, and we form ATP into AMP, what ends up happening is we have AMP plus pyrophosphate. The pyrophosphate gets immediately broken up into two inorganic phosphates. And that's exactly what drives the reaction forward and keeps it at AMP. So that's just what I'm trying to say here, is that we generate this pyrophosphate, and then it immediately breaks apart into two organic phosphate groups. And so, again, as I mentioned before, why we use tagged sugar nucleotides for biosynthesis, they're irreversible because of that pyrophosphate, as I mentioned right here. That's what drives this reaction forward. And then there's favorable enzymatic interactions, so binding energy is also another thing that makes it favorable. Obviously, you have the UDP attached to it, so it's going to allow it to power this whole thing and keep it going. And then there's reactivity. The UDP is also a very good leaving group. It can just stay as UDP. And then, again, it creates separate pools, so it allows for different concentrations to occur within the cell. Okay, so now we're actually forming now that we formed our substrate, our UDP glucose, what we're going to end up doing is forming glycogen. Now, the way we do it is it's not just UDP glucose monomers coming together and forming like Lego bricks, and then you form this huge glycogen molecule. You need to start off with a base, and the base is going to be glycogenin. And glycogenin is a protein. It is not a sugar, it is a protein, and it contains a tyrosine molecule. Now remember, tyrosine molecules have the hydroxyl group, and that allows them to become phosphorylated or participate in glycosidic bonds. That's exactly what happens here. So our UDP glucose, right, it's going to be powered by that UDP that's attached to it, and the tyrosine is a bit nucleophilic, right? And we have such a good leaving group that this reaction is actually favorable, right? So the OH from the tyrosine is going to attack the anomeric carbon because it's connected to two of the oxygens, and it's going to form our chain, right, of the glycogenin plus attached to the monomer. And then from there, it starts getting added like Lego bricks. Now, I wanted to briefly emphasize that we're using a different enzyme, right? So we're using this glucosyl transferase activity. This is separate from glycogen synthase, and we'll talk about this, and I'll talk about it in the next slide. But this one is going to transfer our UDP glucose and put it into the tyrosine. So that's what this first enzyme is doing. Now, the second one is our chain extending activity, where we now are dealing with multiple sugars being combined in glycosidic bonds. So we're obviously not going to use the glucosyl transferase because we're not dealing with proteins anymore. We're dealing with just extending sugar molecules, sugar chains. So that's these two enzymes that help with the initial generation of glycogen. Now, when we're talking about branch extension, right, we're going to be using glycogen synthase, and it's basically like a two-part enzyme. It has two functions inside of it. So glycogen synthase first needs, and you have to memorize this, needs a primer of 8 glucose residues before it can be used. So we have to undergo this chain extending activity 7 times, right? Because we need to add 7 glucose molecules to the one that we already incorporated. So this occurs 7 times, this chain extending activity, and then the glucosyl transferase activity occurs once. So that forms our primer now. Now glycogen synthase can come in and say, okay, we can start adding glucose monomers quickly. And remember how I mentioned that it has 2 functions, right? So it transfers, the first function is it transfers the UDP glucose to non-reducing ends of the glycogen branch. Remember back in lecture 7 when we were talking about sugars, how I was telling you that non-reducing end, that's where you have the glycogen synthase activity, the phosphorylase activity, it happens from the non-reducing end. So we end up adding to the non-reducing and extending it that way. Or extending it the other way, towards the reducing end. My apologies. And then for glycogen synthase, we end up forming alpha-1,4 linkages, right? So that's the first function of glycogen synthase, is that linear chain extending activity. Whereas our glycogen branching enzyme, that's the second part of this glycogen synthase, where it'll take essentially the terminal end, which is 6 or 7 residues, and that's from the non-reducing end, and basically take them towards the reducing side of this glycogen polymer. And what ends up happening is it forms a alpha-1,6 linkage, and then from there, that's exactly what gives us the branching activity into different branching activity and forming this very complex glycogen molecule that's able to neatly pack together. And then, does anyone have questions about glycogen synthesis, anything like that, before we move on to glycogenolysis? Yeah.
Wait, so glycogen synthesis just extends the branch, and the branching enzyme creates a new branch?
Yeah, so glycogen synthase, I like to think of it as, actually no, they're 2 separate enzymes, my apologies. Glycogen synthase extends the chain activity. Glycogen branching enzymes take the basically chain you just created, clip the end of it, and take it to the very beginning, the reducing end. Does that make sense? Okay. Any other questions? Yeah.
Can you repeat why we use glycogen synthase instead of continuing to use
Glycogen synth. Oh, so can you repeat that question one more time? Sorry.
Like, because
Mhm.
glycogen synthase just adds monomers using the same UDP glucose, why don't we just continue using glycogenesis?
I think I like me making up a BS answer to like try to cover me is like it has the active site just can't accommodate that very long chain or whatever because I don't know. That's just what I would say, but we know that we're starting from a protein, right? Protein's going to be its own shape, whatever. It's going to be a different size. And so because we're starting from a protein, it's going to have different properties and it's going to interact differently with the glycogen synthase. That's why we don't have the glycogen synthase just come straight in and then start extending the chain. We have to use a different enzyme to accommodate for those different features of the protein. And in terms of your question, which is, well, why doesn't it just keep using that chain extending activity? It could just be that that enzyme specifically will not work anymore because the chain is extended so far and it's like out of the active site or something like that. And it just switches. Does that answer? Okay. So then moving on to glycogenolysis. So we're breaking the glycogen down. Let's say that we have low glucose levels, right? And we have high energy. This is another scenario, right? We have, wow, okay. Oh, that's a bad idea. Okay, let's just say we have low glucose, right? So we need to put glucose into the blood. And let's say we have high energy, right? Wait, give me a sec. We have low energy, sorry. Low glucose, low energy. So if we have low glucose, we need to make glucose into it, but we have low energy. So we can't just go through gluconeogenesis, right? We can't just form the glucose from that pathway. So what we have to do is break down the glycogen that we have in our body. Does that make sense? And this is prevalent in muscle cells. Remember, muscles at first use oxidative phosphorylation or they have the glycogen stores whenever they don't have the energy. Because our muscle cells, our contractions, we use ATP. So if we use up all the ATP, we can no longer do gluconeogenesis because the ATP is gone. But we still need that glucose. So how do we take it? We take it from the glycogen breakdown. And that's the whole prevalence of glycogenolysis. So for the first part, we have the first enzyme that we're dealing with is glycogen phosphorylase. It removes the terminal residues and releases glucose 1-phosphate. So it releases it from, if I'm not mistaken, the non-reducing ends. So it takes them from that end and forms glucose 1-phosphate. And then it just has to undergo the mutase enzyme to form back to glucose 6-phosphate. And then it can be used and converted into glucose via the glucose 6-phosphatase. And one important thing to note about glycogen phosphorylase is that it stops 4 residues from the branch point. And they're colored differently here for another reason, but I'll explain that in a little bit. But basically, it stops here essentially after when there's 4 left. And then we have the this is where I was getting confused. So the debranching enzyme has 2 inside of it, 2 inherent activities. This is what I was trying to allude to. So the debranching enzyme has the transferase activity and the glucosidase activity. The transferase, as you might imagine, it transfers something. So it's going to take the 3 from the non-reducing side, it's going to take those 3 glucose monomers and transfer them to the non-reducing end of a long chain already, if that makes sense. So we're going to take this, we're going to cut this bond between the first and the second monomer from the branch point, and we're going to basically transport it all the way to the end, the non-reducing end specifically. And then when we have this 1 glucose monomer left, what's going to end up happening is we're going to use this glucosidase activity, and it's going to remove this alpha 6 bond, and it's going to release just a free plain glucose. Not a glucose phosphate, anything. It's a free glucose. Does that make sense? Does anyone need clarification for that? Okay. And then here's another regulation step. We'll talk about this. This one is also very confusing as well. So first thing I want to orientate you for state of like matter of fact things is pay attention to glycogen synthase, right? It is hydroxylated when it's active, and then it's phosphorylated when it is inactive, right? So hydroxylated, active. When it's phosphorylated, it is inactive, right? And the same is true for phosphorylase, right? When it's hydroxylated, it's inactive instead, right? So in this case, we see this like dual thing. If it's hydroxylated, it favors the glycogen synthase activity, right? Because this is active, but the phosphorylase is inactive. Whereas if we phosphorylate both of these enzymes, if we stimulate that kinase and it phosphorylates both of these, we see that the glycogen synthase is inactive, but our phosphorylase is active. So I just really wanted to emphasize the matter of like fact things. If it's phosphorylated, we want to phosphorylase things, and so therefore we want to break down glycogen because that's what phosphorylase does. But if we have a hydroxylated state, the glycogen synthase is activated and the phosphorylase is inactivated. So just know that that's a matter of fact. That's always true. Now, in terms of regulation, this is where it gets a little bit confusing, right? So we'll stick with specifically glycogen phosphorylase, right? This right part of the diagram. So it is active, obviously phosphorylated, all that stuff. So it is activated by glucagon and epinephrine. First, we'll talk about glucagon. This is what I was talking about. We have low blood glucose levels. Glucagon is secreted in response to that. But let's say we also have low energy, so we can't just do gluconeogenesis. So glucagon is stimulating phosphorylase kinase, and what that does is it phosphorylates the phosphorylase. It basically phosphorylates the enzyme, and it'll activate it. And when it activates phosphorylase, it's going to break down the glycogen stores. And that makes sense, right? If we have low energy, low glucose, we want to form glucose, but the only way we can do so is breaking down glycogen. So that's the way to physiologically just think about it. And then with epinephrine, calcium, and AMP, I don't want to confuse y'all too much, but essentially remember skeletal muscle, right? We know that the way it does the contractions, it does so is through a huge influx of calcium, right? So calcium, okay, good. So calcium ends up having a huge influx inside of the sarcolemma, the cytoplasm of the muscle cell, and that binds to troponin, changes the conformation of tropomyosin, and then the tropomyosin binding sites can bind onto the myosin heads, and that allows for cross-bridge formations to occur. So now our muscle is activated, it's contracting, and because we have the influx of calcium, we need to make sure that we're generating ATP. And again, if we have prolonged muscle contraction, our ATP levels are going to drop so much, and we have that state again that I was talking about: low glucose, low energy. So we have to stimulate glycogenolysis, breaking down glycogen, and that's why this would activate it. I can go over it again if you guys want, but if not, it's totally fine. Yeah? Okay. So in muscle cells, we know that they contract when we have an influx of calcium. When an influx of calcium occurs, it binds onto troponin proteins in the muscle. When it binds onto the troponin proteins, we know that changes the conformation of the tropomyosin proteins, and it exposes exposes the binding sites to the myosin heads. So now we have a condition where calcium is high, it opens these binding sites, and myosin can bind onto it. What this means is now we can have cross-bridge formation and power strokes, which just means that our muscles contract. And we know that requires ATP, a lot of ATP. And so if we have prolonged muscle contraction, our ATP levels are going to drop a lot. And what ends up happening is when the ATP levels drop and we're running low on glucose because we're exercising for too long, then we have to create glucose somehow. And if we have low levels, we can't just create it from the ground up. We have to take our internal sugar stores, which is glycogen. Does that make sense? Yeah. Okay. And then that's the phosphorylase. Now talking about the glycogen synthase, right? So we know that glycogen synthase is active when it's hydroxylated. It's inactive when it's phosphorylated, right? So we'll first talk about the condition with insulin, right? Insulin says that we have high glucose levels and that we need to get rid of the glucose somehow, whether it's glycolysis or whether it is through forming glycogen, right? Now, what happens when, we have, for example, high energy, right? We don't want to go down glycolysis. Well, like I mentioned, we have to go through the pathway of glucose, and we have to get rid of it and go through glycogen synthesis. So that's exactly what this is doing. This is a phosphatase protein, which it will dephosphorylate our glycogen synthase, and now we'll form these hydroxyl groups that will activate it. And so that's why we see insulin stimulate this phosphatase protein, so it's going to favor the hydroxylated state. But additionally, it also inhibits GSK3. GSK3 is a kinase, meaning that it will phosphorylate our glycogen synthase. So when we inhibit GSK3, we're disrupting the ability of glycogen synthase to be phosphorylated. So all I'm trying to say is when insulin inhibits GSK3, our glycogen synthase remains hydroxylated longer. Does that make sense? Because we're inhibiting the kinase activity. That's what that is, that regulation step of insulin. Does that make sense? Okay. So that's insulin. Now, in terms of glucose, obviously we have high glucose levels, right? As I mentioned, it's like it works in tandem with insulin. Glucose is going to stimulate, again, this phosphatase, and it's going to form this active glycogen synthase, right? And same thing with glucose 6-phosphate. In order to go through glycogen synthase, remember that we have to convert glucose into glucose 6-phosphate, and then from glucose 6-phosphate to glucose 1-phosphate, then we turn it into UDP glucose. And so these two are going to just work in tandem with insulin as well, and these three obviously are going to activate glycogen synthase. Now, the thing that will inhibit this pathway is glucagon epinephrine. And the way it works is if we have low blood glucose levels, we obviously don't want our internal glucose levels to go down by going to glycogen, right? We don't want to form glycogen when we actually need the glucose in the rest of our body. And so glucagon is going to inhibit the phosphatase activity. What this means is that glycogen synthase stays in the phosphorylated state for longer. Does that make sense? So when it's in the phosphorylated step, then what ends up happening is our phosphorylase is active, and we end up breaking down the glycogen. Okay, does that make sense? All the regulatory steps for this? You just have to keep it separate from energy and glucose, and then you have to play with the two conditions. When you do, it starts to make sense, right? Yeah.
When I was like looking through the book, they had like a structure at one point that kind of showed these two, like the synthase and phosphorylase, like as part of a protein kind of like complex together with the PP1 on it. So are they kind of like adjacent to each other, or are these separate?
I never heard of them being together. The only ones I've heard being together are PFK2 and FBPase2. So I would think of them as like perhaps close to each other, but not necessarily like connected in the amino acid sequence, if that makes sense. And yeah, and I mean, I'll stay around for questions, but I wish you guys luck for the exam. I will post the recording as well, so if you guys need to like just listen to it a couple of times, it'll be posted as well.